Thank you all of you for joining such a early in the morning. I think it is 9 sharp now, and we have a session ahead. So we are trying to do the things at the earliest possible. So our our YouTube live streaming has also got started. In the morning, I think it is 9 sharp now, and we have a session ahead. So we are trying to do the things. So now we are live on YouTube also. So good morning to the delegates, speakers, and resource persons of the day, and uh, I heartily welcome all, one and all, Mrs. Indira Vittal from New Zealand, and Dr. Ramakrishnan from China, and other participants. Now we can start formally inaugural session. And in this inaugural session, I would welcome, because as the registrar madam is having some important meeting, so we can ask her to address first, and then we can start inaugural. Professor Naseem ma'am, over to you. Can you please give inaugural address for us?
Esteemed Dr. Ramakrishnan sir,
thank you, ma'am. Thank you.
From China, and Indira madam, she's from New Zealand, convener of this international conference, Dr. Haleem Khan sir, from head of the botany department of Telangana University, Professor Vidyavardhini madam, and Professor M. Aruna madam. Professor Vidyavardhini madam, she is Dean CDC of our Telangana University, and Professor Aruna madam, the principal of Education College, and other delegates, participants of this event. A very good morning to all of you. I congratulate Dr. Haleem Khan sir, the convener of this event. In this pandemic situation, he has organized a number of conferences, which was quite fruitful to all the participants. International conference. I'm on the way to Hyderabad. I'm facing the network problem also. So I wish you all the best. Thank you.
Thank you, ma'am. Thank you for your kind words. Now I request Tippeswamy sir, Dr. Tippeswamy sir. Can you unmute yourself, Dr. Tippeswamy from Davangere University? He has joined us. Sir, good morning, sir. Are you there, sir? Okay, we'll start with Indira madam. Indira madam, can you give us something the blessings in the fine morning with a fine meeting event?
Thank you for the opportunity, Haleem. I'm really thrilled, and I really feel it's a blessed day. Thank you for that. If you don't mind, I can start with Ganesh prayer, and then we will start. Being an Indian, and I was a colleague for Haleem in Nizamabad Giriraj College, and from there we still continue to be professionally connected. So this has given opportunity for this one. So Shuklam Bharadaram Vishnum Sesivarnam Prasannavadhanam Dhyayet Sarva Vignopasantaye Agajanana Padmarkam Gajananam Harnisam Anekadantam Bhaktanam Ekadantam Upasmehe Saraswati Namastabhyam Varade Kamarupini Vidyarambam Karishyami Siddhir Bhavatu Me Sada. May the Lord Ganesh and Goddess Saraswati guide us in our educational pathways. Thank you for that. And it is really a privilege to meet all these eminent people through Haleem. It's a unique opportunity, and I'm looking forward to this event. Thank you for this opportunity, and may this be blessed by all. Thank you.
Dr. Tippeswamy, sir, are you there in the meeting? You have joined, sir. Okay, anyhow.
Yes, sir.
Sir, you unmute and you switch on your camera also, sir. He has become our best friend for the department because from last meetings he has gone so close associated, and I feel that he can give some address in the inaugural session.
Sir,
It's camera on is good, sir.
Yes, sir. Yes, sir. 1 second. Hold. I will help. Good morning, everyone. It's a very immense opportunity to give inaugural means something like a good opportunity for me to encourage the art Aleem Khan for giving this opportunity. So last time, unfortunately, I wouldn't present successfully my talk because of the network issues, and also I was busy with some of the academic issues. So thank you, sir. Thank you for the opportunity. I wish all the postgraduate and graduate students please make use of these sessions. These are all very useful. I think you people won't get like this of information at the single platform. Kindly, my request is make use of the what we call our speakers work and respect the work, and inculcate all the whatever the information is there in your fields. So thank you, sir, Aleem sir. Thank you for the opportunity. All the best.
Yeah. Anyhow, you are having a session, Dr. Ramakrishnan. Even though you can speak few words for the inaugural session. Unmute yourself. Unmute yourself.
Very good morning to all. So thanks to Dr. Haleem sir for organizing this international conference. And also special thanks to Indra madam for special prayer to the conference. Thank you very much, madam. It is a very pleasure to giving the talk from the China to the Indian students, especially from the botany department. Basically, I am also botany student. When I did a graduation in the botany, then I moved to biotechnology. So all my professor, well-wisher, they are working in Tamil and the botany department. So I am very happy to be participate in the botany department conference. Yes, sir. I can continue, or maybe if anyone want to speak, then later I can talk. No problem, sir. Yeah.
Thank you, Ramakrishnan. Thank you, the speakers earlier. Srinivas has joined with us. I will make him the co-host. Dr. Srinivas, can you please speak few words in the inaugural session? Dr. Srinivas, our colleague in the department, Dr. Srinivas, he was not doing well yesterday, but today he was said he's fine. Dr. Srinivas, can you speak few words in the inaugural session? Srinivas, I have made you co-host. You can speak, Dr. Srinivas. Anyhow, meanwhile, I request Professor B. Vidyavardhini Madam to give her address in the inaugural so that we can begin the session in time.
Good morning, everyone. Welcome all to the second International Conference on Emerging Trends in Life Sciences on Virtual World, organized by the Department of Botany. And we are immensely grateful to our registrar, Professor Naseem ma'am, for inaugurating the session, and though she was busy on her schedule. And thank you, Haleem Khan, for organizing this. And thank you, Indira ma'am and Dr. Ramakrishna for speaking on this inaugural session. And I hope all the students and all the delegates who are participating will inculcate various aspects in plant sciences, where you will understand certain things which you might not have read or you might have not have come across. We have got eminent speakers from different parts of the world, and we have got from different parts of India who are going to focus on all the aspects related to your studies, as well as not concerned to your studies, but something which you will understand about life sciences. So once again, I welcome everyone, and I thank Haleem Khan for giving me this opportunity. Thank you and God bless.
Aruna ma'am, good morning. Professor Aruna madam of our department has joined the meeting. Ma'am, can you listen to me? Aruna ma'am? Ma'am, I made you co-host. Can you speak? I hope she dropped. She got dropped from. She's trying to join again. So overall, on behalf of Department of Botany, Telangana University, Nizamabad, it is proud privilege from myself, Dr. Ahmed Abdul Haleem Khan, to welcome you one and all for the 2 days international conference on emerging trends in life sciences. So this is a virtual mode international conference to be conducted for 2 days. That is today, 28th and 29th of January 2021. So we have got 4 technical sessions today after the completion of inaugural session. The inaugural session will be from 9 a.m. to 9:30 a.m. And after that, we'll have a technical session with 2 eminent speakers. One is Dr. M. Ramakrishnan from Zhejiang ANF University, Hangzhou City, Zhejiang, China. I can't pronounce things. So he'll be addressing us about his invited talk, and followed by that, we'll have Professor Vasuki V. Belawadi, sir, from Department of Entomology in University of Agriculture Sciences, GKBK. He'll be dealing with pollination and ecosystem service that is taken for granted. And then technical session 2 will begin from 10:30 to 11:30 a.m. And where we can listen to Ms. Indira Madam. She'll be addressing us on the role of science in our daily life and learning. Then followed by that, we have Professor Maman Daniel, ex-dean faculty of science, MS University of Vadodara. He will be speaking on business opportunities in field of medicinal plants. Followed by that, we have a technical session 3 where we can see 11:30 to 12:30. We are going to listen Dr. Ashwin Kumar Dikshit sir from Guru Ghansidas University, Chhattisgarh. He'll be speaking us on the immunomodulatory herbal medicine. And then we are going to witness one more speaker from Australia, Royal Melbourne Hospital staff unit manager, nurse staff unit manager, Hemalata Uppu. She'll be speaking on global effects of the pandemic. What is the current situation? We need to get alarmed. As the vaccination schedule has arisen, we need to know much more details about what the paramedical is having their experience in the field of pandemic. Then we'll be having 2 more speakers in the technical session 4, where we are going to end today's session. Dr. Mayur D. Nandikar will be speaking from Shirwal. It is Nauroji Cooperative Center for Plant Research. He'll be speaking on Indian Comoliniaceae, diversity and endemism, and followed by that last talk will be from Dr. Ram Prasad sir. He's from Mahatma Gandhi Central University, Motihari, Bihar. East Champaran. So with this few words, I hope that whatever the today's technical sessions are there, today's technical session will have the amalgamation of both the speakers from national and international from India and outside the India. National and international speakers are there in this 4 technical sessions, where we will be listening the various aspects of the science and life science progress, as well as the current situation, that is the pandemic. So the department, about the department, when we speak, the department has started in the year 2006, when the university had the inception of university. And presently we have the 5 faculty members in the department, with 2 professors and 1 assistant professor and 2 academic consultants. So the department has got research supervisorship for all the faculties in the department. We are presently with 14 research scholars working in various fields of applied field of sciences, like plant physiology, and then hydrobiology, and algal biotechnology, and mycology. Like various fields, the research is going on, and more than 12 members has completed their PhD. So the department, slow and slow, we can see the growth of our department. We have got the 3 research projects by different funding agencies, and we have got 2 Inspire Fellows who worked in our department, and 1 Rajiv Gandhi Fellowship research scholar has completed her PhD. And then presently we have 1 Kothari Fellow who is working under guidance of our able Professor B. Vidyavardhini Madam. And the department has got the various support for various events in the department. Like we have conducted more than 4 science academies workshop, and we have also conducted more than 3 national conferences, and offline mode. And we have also recently completed the Indian Academy supported national level science academies lecture workshop. And also we have recently finished a virtual mode international conference and national conference, and we have got good grade from the NAC accreditation. Also, the department has actively participated in various pursuits, academic pursuits of the university. With these few words, I would like to request Aruna ma'am to speak the gathering. Aruna ma'am, your co-host, you can speak now. Aruna ma'am. Good morning, ma'am.
Good morning, sir. Am I audible?
Yes, yes.
Good morning to all the delegates present over here. And I welcome all the delegates and participants from all different states across the country attending for this 2-day international conference on emerging trends in life sciences. And we have invited resource persons from all over India and also other countries like China. And we will be having technical sessions as briefly explained by Dr. Haleem Khan sir. And I hope that all the delegates and the participants will be present for 2 days and be benefited by the lectures delivered by different people. Thank you. Thank you, sir.
Thank you. With this, as the inaugural comes to an end, I request our speaker, as Ramakrishnan is very busy in his academic pursuits. So I request Dr. Ramakrishnan to begin his presentation. It is sharp 9:20, and you can finish in time, and you don't have any hurdle, I hope. So Dr. Ramakrishna, you can start sharing your screen and you can begin your presentation. Technical session 1 begins now.
Can I able to see my screen?
Yes, yes, we can able to see you. Yes, yes.
I think it's okay. Thank you very much for the Kalim sir for giving a very nice introduction about Telangana University. So it is very happy to give the presentation in this university. It seems the university is with the NAC committee and everything, they give congratulate the university. Okay, sir. So shall I start now?
Yeah, yeah, you can.
Okay. So basically, I was in the Zhejiang Agriculture Forest University up to December 31st. Now I moved to Nanjing Forest University. It is one of the prestigious university in the Nanjing forestry field. Now I am here working as an associate professor. So I am working in the Bamboo Research Institute. Today I am going to talk about functional analysis of Moso bamboo transversal elements. So, but in the sir mentioned in the presentation, the invitation letter, I will talk about regarding some partial in the partial millet phosphorus use efficiency, but that was my PhD work. Then I thought why, because in the partial millet, I could not focus too much about molecular work. So this is a complete molecular work. So the students will get benefit attending my talk. So why we want to study about bamboo? So bamboo, we know the bamboo sustainable goals for contemporary world seeks a viable solution for interconnected challenges, particularly in field of food and energy security and climate change. As you know, the major problem in India, not only in India, all over the world, energy and climate change. But the bamboo is one of the versatile plant species on earth. It is an ideal candidate of bioeconomic for meeting some of these challenges. Bamboo can be used for energy production, food security, and climate change. With its potential realized particularly in the industrial sector, countries such as China are going extensive with the bamboo development and cultivation to support the industrial uses. This includes timber, fiber, biofuel, paper, food, and medicinal industries. Having better adaptation to wider environments than to other grasses, bamboo is an ecological viable choice. So it's viable choice and can help to restore the degraded lands and mitigate climate change. The application, the main advantage of the bamboo, it can grow in the wild climates like highland to lowland, cold to heat. So it can grow wherever we can grow it. Yeah. So this is some of the application of the sustainable for we can develop bamboo furniture, bamboo toothbrush, bamboo cups, bamboo paper bags, and some of the sandbars, clay pots. These are the sustainable development. But the main application of the bamboo development, biodiversity and tourism. So the bamboo forest is very, very important for the biodiversity and tourism. Then the value-added product for the tissue paper, food and medicine, fiber cloth, biofuel, renewable energy, charcoal. It is a main important for the fertilizer. Then furniture and construction, then computer, ornamental and handicraft. There are, so far they have reported, we can produce more than 3,000 products from the bamboo resource, using bamboo resources. So bamboo is one of the crop species. Okay, which is become amenable to genetic improvement to its long breeding cycle, perennial nature, and mono. Because the problem in the moso bamboo, we don't know when it will produce flowers. Maybe it take 100 years or maybe 150 years, sometimes 10 years. So according to the temperature, the bamboo will produce a flowering. One of the commonly used species is a moso bamboo is a potential candidate that qualify as industrial bamboo. With the whole genome information released to genetic manipulation of moso bamboo offer tremendous potential to meet industrial expectation either in quality or in quantity. Further, bamboo cultivation can expect several natural hindrances through biotic and abiotic stress, which needs viable solutions such as genetic resistance. So, although bamboo can grow in the wilder climate, but still we need to develop the bamboo for some of the resistance like biotic stress or abiotic stress. taking a programmatic view of these future requirements. So we have been studying most of bamboo genetics, genomics, transversal elements, and epigenetic modification to try various implications in meeting industrial and cultivation requirements. So there are three concepts. How can we develop the circular economy? Because the many country now they are focusing circular economics. Bamboo can be used for circular economy and sustainable development. One is biology, molecular genetics, then quantitative genetics, then biochemistry. If we combine all three, we can develop a circular economic to develop the sustainable development. So there are many applications are available to develop the bamboo genetic improvement, but the most problem in the one is characteristics of growth and development, the genome sequencing, alternative placing, gene family functional analysis, then active transposon, mutant library, combined transcriptomics, proteomics, biotic and abiotic trace, flower development, shoot and rhizome development, then gene editing, in vitro somatic embryogenesis using gene editing, gene transformation. These are the methods we can follow. Yeah. So based on this, we have published one review in Food and Energy Security that impact factor 5.5 genetics and genomics of most of bamboo, current status, future challenge, and biotechnology opportunities towards sustainable bamboo. It's a very interesting review. I may request to the students, if they are interested, they can go through this review. We given complete overview of the most of bamboo genetics and what are the challenges in the bamboo development. So now coming to the main part of in the talk. So we have been working in the transposons. As we know, transposons, many people thought in the 1970, even up to 2000, many people thought it's a non-coding region, and it is it's a waste elements in the genome. When the Barbara Clinton, she reported the transposal elements, her colleague thought she is a mad. Nobody recognized his his report, because they thought transposable elements is a jumping gene, it is a mobile genetic elements, it don't have any functions. Now, but after some years, they found some of the interesting mechanism in the transposable elements, and now many people are working transposable elements. It is now the complete our epigenetic mechanism, even the human behavior, it's associated with the transposable elements. So because in the transposable elements, it can control the complete our behavior, the characterization, our thoughts, everything is associated with the transposable elements. So we are working on, but it is a very difficult to study transposable elements, because it will always jump in the genome to one place to another place in the genome. And it has some mechanism, how it is activating in the gel and how it is controlling the cell mechanism and everything. So to study this one of the mechanisms, in our previous work, we identified two elements. One is BPMR1 and BPMR2. This is the two transposon elements, but we don't know how it is works in the gel, in the cell. So we studied the nuclear export signal of transposase affects this transposase of this signal affects the transposition activity of mariner-like elements of most of bamboo. So generally, there are two types of transposons. One is DNA transposons by DNA mediated cut paste mechanism. Then another one is retrotransposon. That is a class 1. We can call it as RNA transposon. That is copy and paste. In the DNA transposons, it will copy and paste in the genome within the nucleus. It won't come to the nucleus cytoplasm. But whereas the retrotransposon can move, first it will transcript the transposon into the cytoplasm. Then cytoplasm will make the circular chromosomal DNA. Then again it will come to the genome. Then it will integrate. So that is a mechanism. although DNA transposon and RNA transposons are transposable elements, but the mechanism are different. But we are working on DNA transposon, how it is jumping in the genome. So the most prevalent DNA transposon families in the genomes is TCL. There are many families like a gene family, like a plant, taxonomy. We have, I think botany students, they know about the plant taxonomy and the genus, species, everything. Like that, in the genome also, we got many families. The DNA transposon itself, we got many families, DNA transposon families and RNA transposon families. Among many families, mariner-like elements have relatively simple structure consisting mainly of terminally inverted repeats. So it is a 10 to 40 phase per long repeats. It gets both end of the genome. Then it merely encodes the transposase enzyme responsible transposition activity. Structurally, they contain DNA binding domain and catalytic domain. So this transposase enzyme is responsible to cut the transposon. After cutting the transposon, then only they can go and integrate, locate in the another place of the genome. So basically, this is some picture about the transposable elements. Okay, so we can see the this is the class 1 retro transposons. It's non-LTR, LTR, TRS. Then in the this is a subclass, subfamily. Then the family. In the family, we have many of these LTR retro transposons. Whereas in class 2 DNA transposon, we have the double-stranded DNA transposons, circular DNA transposon, circular DNA krypton, and double-stranded DNA. Then we have the TCL mariner, and TCL mariner we have TCL1 and MOSO1, and in the Kachek, this we have the another type of transposons. But these all the families we could not find in a single species. Maybe in the bamboo we found this, but in the rice we found this. So then we are interested in these TCL1 elements. Yeah. So what is the importance in the transposable elements? The problem is we should know at least 10 things about transposable elements. Why it is important in the genome? The first one is all the transposable elements are not uniform. It is different from shape to species to species, and the size also different. It is not randomly distributed in the genome. It is not randomly distributed in the genome. Then the source of the mutation, the main source for the mutation is the transposable elements. Because transposable, wherever it sits in the genome, it will create the mutation. Then the genome arrangement. For example, this genome sitting here, this genome is sitting, then the arrangement also will change. So genome rearrangement. Then the expression and repression. The transposable elements, they can also activate or deactivate the gene expression. Then affect the germline and somatic embryogenesis in the human genome. Then not just for transposition, it is not a transposition, it also improve the immune response in the genome. Then creates a lot of non-coding RNA like the circular RNA, long non-coding RNA, siRNA, microRNA. There are so many RNAs derived from transposable elements. Then modify the regulatory networks. So many non-coding transposable elements, they are regulating the complete genome, such as regulatory pathway, metabolic pathway. These are all the major pathways. Then requires specialized tools. The problem in the we want to study the transposable elements, we need the specialized tools. So far, what we have, the technology is not enough to study the transposable elements. Yeah. So in the moso bamboo, because moso bamboo, we know it is a monogod species, although it is monogod, but it is a non-timber, it is like a tree. It will produce a flower after 100 years or maybe 50 years. It's the flowering mechanism irregular. Irregular means we cannot predict when it will flower. But it has 63% of transposable elements correlating with the genome size, playing and important roles in the bamboo genome. Because in the TA, the transposable elements are not randomly disturbed. It always waits to propagate through the sophisticated interactions. These results together suggest that the TE proliferation might be a major driver of bamboo development. So because the most of bamboo has 60%. So based on this structure and their sophisticated interaction, scientists they concluded TE proliferation might be a major driver for bamboo development. However, TE function in most of bamboo have not yet been completely studied. So we can see there are, because there are different types of bamboos, but this is a Moso bamboo. So Moso bamboo we have 45% of the LTR. This is only for LTR, and the Bonia we have 63%, and the Gudwa we have 45%. Then Oliera and the Radia. These are the different bamboo species. But among these bamboo species, LTR is a major source of the bamboo. To use them as a potential tool for genetic investigation in mouse of bamboo, there is a need to document the efficiency of these elements. There are several factors that determine the transposition efficiency, because when the transposition want to jump one place to another place, there are so many factors. We cannot say it is only because of this factor, because of this factor. There are so many factors affecting the transposition. Transposition means when the transposition moving one place to another place, that is called the transposition. Okay. Such as internal, external factors. Internal factors are primarily structural features such as TAR, sub-TAR, and transposase sequences. This is a enzyme sequences. Like that, there are so many factors that can control the transposition efficiency. There can be several external factors also, such as cellular environment, quantum and duration of accumulation transposase, age and metabolic state of the cell, and the abundance of active TEs in the cells. So in our study, what we have done, so we studied the PPMR1 and the PPMR. These are the two MLE transposable elements, TNA transposable elements. We already cloned from Moso bamboo. Okay, then it has some nuclear export signal, but not nuclear localized signal. So the signal is very important when it is going to from the nucleus, from cytoplasm to nucleus, then it should move to nucleus to the cytoplasm. To understand this mechanism, okay, so we have conducted two experiments, fluorescence and excision frequency assay in the yeast system. For this, 3 NEU mutants were developed from each of the MLEs by site-directed mutagenesis. As you know, that is a cloning method to site-directed mutagenesis. In the fluorescence assay, the mutants NEs 1 and 2, 3, along with the wild type NEs 0 were co-transformed into the yeast system. So we used the yeast system along with the fluorescent proteins. The fluorescent proteins to localize, to see the where it is a transposition enzyme located, whether it is a cytoplasm, whether it is in the nucleus. So to indicate that, we used the fluorescent proteins and enhanced yellow fluorescent protein and enhanced cyanon fluorescent proteins. To differentiate the protein localization under the TE influence, ECF alone was fused to wild and mutant NES, and domains are N terminal or C terminal. I think it's okay. Yeah. So this is amino acid mutation in N. So this is a sequence of the transposase. This is a signal. So this signal is very important for the transposase. This is the wild type. Then this is the strong NAS, weak NAS, intermediate NAS. This is a mutated. Here we mutated glutamine. The amino acid glutamine we mutated into leucine. So leucine. In the weak NAS, we mutated the leucine into alanine, then leucine into alanine. This we have mutated. So like that, we have mutated in the PPMR1 and the PPMR2. So then according to there is a database to identify the NE signal in the amino acid sequence. According to the score, we predicted it is when we mutated glutamine to leucine, that is approximate score is 1. Then like that, we have identified the weak, whether it is weak or strong or intermediate. So this is it's a wild type almost near to 1. But in the mutant strong NAC score 1 and it is weak NAC 0 and intermediate it is 0.5. Yeah, this is amino acid sequence of transposase 1, ppmr1 and ppmr2. So this is each is coding for the amino acids. Now I think that maybe students now they may understand what is a transposon. So this is the ppmr transposon. This is a complete transposase. So you can see this is a left TIRS, right TIRS. This is a terminal inverted repeats. So the ppmr transposon, to study this, we constructed a non-autonomous transposon like that without transposase enzymes. So this is you can see the left and right. This is exon and DST. So we removed all these and we constructed the ppmr non-autonomous transposons. So using these transposons, so we have transferred this within the this in the yeast system. We can see now. We think you will understand. This is a methodology how we did. So in the ppmr transposase, it has wild type and mutant. It has all wild type and mutant transposase. Okay, this is complete. This is the class ppmr1 and this is the ppmr2. So we have done individually for all these transposase enzymes. This is a transposon, non-autonomous. So this transposon cannot move without these transposase enzymes. So what we have done this, we transferred this BPM non-autonomous transposon and the transposase in the vector. Okay, we transferred in the PW89A PPMR2A vector, and the transposase we transferred in this TPS1 vector. Same like the PPMR2 transposase we transferred in this vector. After transforming into two vector, again these two we transferred into the yeast vector. So yeast we transferred codon with the strain DG2523. So this, after transform these, we added with the ADET revertant frequency. That is, that is some mutant it will. So without this ADET revertant, the ppmr transposon it won't grow. So that means it's lacking urea. It is urea and histidine and galactose. This without these elements, that yeast cannot grow. So like that we have added this CSMIDQ urea 2-chloro. So like that here also we added these two co-transformation with this. This is a methodology. I think I hope that the students can understand this. Yeah. So based on this fluorescence assay revealed that the blue fluorescence of EC was more intensive than the red fluorescence of YF in the yeast cells matrix. Further, ECF had a wider localization in the cellular matrix, but UYFE was largely located in the nucleus. So like that, we have studied all the mechanisms in the yeast cells. So after that, we see. So the co-transformation vector together with the non-autonomous PPMR transposons and NES locking transposase was used to assess the differential excision frequency of the mutant NES domains. So I think it may be now it may be clear. Okay, so you can see this is a mutant PPMR1 0, PPMR1 NES, PPMR2 2 and NES3. This all three is a mutant. This is a wild type. This is a green channel. Oh, no, this is the yeast cell. So we took confocal images. It is a green channel, blue channel, and these are merged channels. Okay, this is red channel. So you can see when the NES2 we were mutated, the transposase enzymes are very less in the very less in the nucleus. In the cytoplasm, you can it's indicating there are the transposanes are located in the cytoplasm in the nucleus. But whereas in the NES 0 and 1, you can see that there is a complete spread. You can see that it is complete spread. It means the transposase enzyme it is not stable in the nucleus. It is coming from the nucleus to cytoplasm. You can see this is same like the NES3 and NES. This also you can see. So it indicates that the mutant NES2 have the capability to hold the transposase enzyme in the nucleus so that transposase can cut the DNA, then DNA can move to another in the genome of the another place. So this is a very important. We found that Ne2 mutant is have more capability to code the transposase enzyme in the nucleus, whereas in the NES1 it doesn't have that much capability to code the transposase enzyme. You can see this enzyme is completely spread in the nucleus. It means because the transposase enzyme very important in the nucleus to cut the genome. If there is no transposase enzyme, then how the transposase enzyme cut the genome in the nucleus. So, but this signal it's exporting the transposase enzyme nucleus to the cytoplasm. So this is the mutant we found in the yeast cells. Same like this is a N terminal. This is a N terminal of the transposase enzyme. Then we also studied what about C terminal. So we found that the C terminal also that these transposase enzymes were stable in the nucleus compared to these types. Then this is BPMR2 also. We did also BPMR2 like that. This also we have done because BPMR1 and BPMR2 we have that. This is again C terminal. So then after that we found the mean intensity. We also quantified the mean intensity of these transposase enzymes. These okay in the confocal images you can see it is a wild type and this is a mutant 1, mutant 2 and mutant 3. You can see the mutant 2 this transposase intensity is very less because here also the nucleus the enzymes are not exported to nucleus to the cytoplasm. So because of that it is giving the low intensity. Like that we have studied all the mutant. It is C terminal, N terminal, like that. It's many classifications. Now we can see when the transposase enzymes were more holding time, that means the resident time of the transposase were stable in the mutant 2, it increased the transposition activity. You can see because compared to wild type and mutant 1, but you see the transposition here and NES2 and 3 significantly increased the transposition of this DNA. Same like in the BPMR2 NA also it's increased. You can see this is so it is a very interesting result. We found okay when we are mutating this NES2 it has the capability to induce more transposition activity in the genome. So using this mechanism, we can develop a lot of varieties in the crop system, agriculture system. Not only in the bamboo, we can develop abiotic resistance, drought tolerance, and maybe heat tolerance, maybe the fungal resistance or phosphorus efficiency, nitrogen efficiency. So using this transposase mechanism, we can study lot of interesting research in the future. So so this is a conclusion. What is the application of this transposase enzyme in the nucleus? In both MLE, NES1 had the highest excision suppression. Suppression means it doesn't allow for the transposition activity, which was less than half of the excision frequency of the wild type. Whereas NES2 and NES3 elements showed up to 3 times increase the transposition excision in the wild types. This result suggested that Ne is an important regulator of nuclear export of transposase in PPMR elements, and the mutation of NES domains can either increase or decrease the export signaling. So if you want to increase or you want to decrease, you can use this transposase mutant. So we think that most of them NES regulates the transcription activity ability to maintain the genome integrity. So now we concluded that this NES signal is a major player to maintain the genome integrity in the most of them. So this is the first part, and this we published in the mobile DNA. It is around 4 impact factor. This we published. I'm the first author. We published in the 2019. So nuclear expression of transposase, the transposition activity of this. Same like we have done another work that is also published around. This is 4.6 impact factor. That is affinity of terminal invader repeats. So this finding to domain of transposase affects the transposition activity. That also we found this. This is that next paper. We found this how the transposase enzymes coming and finding these regions. Now the first part we studied that how the transposase enzymes that the nuclear export signal of transposase is working. But in the second paper, we found how the transposase enzyme coming and finding in this region, then cutting the DNA, because the transposase enzyme, the cleave the left TRS and right TRS, not here. This is a complete transposase enzyme coding region. Once the transposase enzyme produced in the cytoplasm, then it will come to nucleus, then it will cleave this region and this region. So for that, the transposase enzyme should find with this region. So we also studied how what is the affinity of this transposase enzyme with this region. So that also we studied, and we published that in the International Journal of Molecular Science. So this is for point factor. So this is that basic some mechanism. Also, we are interesting some of the transposase elements. How can we study the marker pest? Because as India, we know India and China, although there are many people are working in the functional genomics, but the variety releasement is very, very less compared to molecular marker. In fact, the great institute, ICR Institute, Indian Agriculture Research Institute, like Andhra University, Acharya University, then GKVK, Tamil Nadu Agriculture University, and the Punjab Agriculture University. There are so many universities, they released variety because of molecular marker, not on based on functional genomics. Even in fact, today, so many agriculture companies, they are mainly working on molecular marker, because that is a way to, so far we can release the variety using these molecular markers. Although the people are working, there are many publications, they are coming in the functional genomics, but the molecular marker, it gets wide application, it gets a lot of benefits compared to functional genomics. That also be interesting, how we can develop any marker using transposable elements. So this is the title of that. This work, development of and deployment of high throughput retrotransposon based markers reveal the genetic diversity and the population structure of Asian bamboo. So this already we know what is application, but that was we studied about transposon. Now we looking interest in the retrotransposon. First talk, the first part was the DNA transposon. Second part was the retrotransposon, RNA transposon. How can we use the RNA transposon as a molecular marker? So molecular marker is nothing but it is associated loci with some particular trait. Like we can say, I think many people they know RAPD, RFLP, AFLP, SSR, ISSR, like that. So this is also one kind of IRAP marker. That is one kind of marker. So retrotransposon play a significant role in the regulation of gene expression, chromosome size and structure, as well as in the genome integrity. Due to their random insertion, behavior interface of retrotransposon can vary significantly among bamboo genotypes. So capitalizing this feature in the retrotransposon amplified polymorphism, that is IRAP, is a high throughput marker system to study the genetic diversity of plant species. So why we are interesting? See, this is a bamboo. You can see the mutation. These are all moso bamboo, but you see the color difference in the moso bamboo. This is we can call it as a internode. This is a node region. But you can see the color in the node region, in the internode region. This is we can call it is a wall. Okay, we call it a slot. This is a wall. You can see the color difference. You can see the color differences. This is a green, but it gets green in the slot. But the rest of things is so. What is the reason? Because this color variation because of the mutation. How the mutation is coming? Because of transposition activity of transposons. So we are interested in what kind of diversity is among the moso bamboo. So we studied that, we developed some of the IRAP, retrotransposon based marker, and we studied some diversity among the Asian bamboo. So we are the first to report. There are more transposon based markers reported from bamboo genome, and particularly using IRAP markers on genetic diversity. As you know, we report a structure based on the bamboo genome for LTR transfer family, TY3 Hypia family, and Copia family, which revealed a total of 98,850 retrotransposon with intact LTR sequence at the both ends. So grouped into the we used some of the CDKit software to identify only this is some NGS analysis. Based on this NGS analysis, we found 13 cluster of retro elements were found with more than 30 LTR sequence with at least one copy having all intact protein domains such as CAG and polyproteins. So total of 16 IRAP primer were synthesized based on the high copy number of conserved LTR sequence. A study using these IRAP markers and genetic diversity and the population structure of 58 Asian bamboo associations belonging to the Pilostoculus. Because in the Moso bamboo, it is very difficult to collect the different bamboo pieces because we don't know who is the father and mother of this bamboo, because the flowering is 100 years once, so we don't know. So we collected only the genus of the Pilostoculus, which is revolted species. So the bamboo associations were collected from 9 different provinces of China as well as from Italy and America. 3-phased approaches using chiralitial clustering principal component and model-phased population structure divided the bamboo association into 4 subpopulations, that is population 1, population 2, population 3, and population 4. All 3 analyses produced significant subpopulation-wise concerns. Further, all the subpopulation revealed admixture of millets. You can see this is admixture of millets. This is a bamboo garden in where I was a postdoc fellow in the Zhejiang Agricultural Forest University. This is some bamboo garden. We got different bamboo pieces. And this is maybe I think it will be very easy to understand. Okay, this is because IRAP marker cannot amplify in the single strategy like RAPD marker, ISSR marker. The strategies are very simple, but the IRAP marker is amplification strategy is very difficult. For example, this is a primer, but this is a primer. It can amplify from this direction to this direction or this direction to this direction. That is a head to head. Then tile to tile amplification. Again, here to here, here to here, or here to here. Then the head to amplification. That means it will amplify here to here or maybe here to here. So this is some of the amplification in the retrotransposon. So that based on this amplification, we can create so much of variation in the amplifications. So these are the species we collected from different regions of China. It is Zhejiang, Anhui province, then the Shanxia province, Hunan province. These are the regions we collected bamboo species. And these are the IRAP marker, but in the, I think it's some people, if in the SSR marker, we have the forward and reverse primer, but in the IRAP, we have only one primer, but the same primer can work as a forward and reverse primer. So these are the forward and reverse primer. We have developed these 13 markers, and we produced, these are the melting temperature and polymorphic information content, then average allele frequency, total number of amblycan in the population. So we have calculated all these tables. So this is some amplification. It's like we can say it's like a random primer, maybe because we want, because when you are using SSR marker, we can get only 2 alleles. One is whether it is present or absent. But in the IRAP, we will get like RAPD banding, but the banding pattern is very difficult to score. So we used to manually score all the primer, but only the master student, they scored this study. They used more than 2 to 3 months to score only banding pattern because it is very difficult. We cannot score randomly. We got to very accurate when the scoring the marker. So we scored only the thick allele, that is a polymorphic allele or monomorphic alleles. Some of these alleles may be discarded because we don't, it may be non-specific or maybe the amplification problem, something. So we scored only this very thick, and it's showing this like kind of bands we scored like that. And we also studied the neighbor joining phylogenetic tree with these IRAP markers. These all 3 markers also very, they are diversity and they are not uniform. So first on these markers, we studied that diversity, and see, this is the 4 type of clusters. We can see that it is cluster 1 and cluster 2, cluster 3, cluster 4, and cluster 5. Then we are interested in how it is scattered in the genome. So then we found that principal components. You can see we have done 3D components. So you can see cluster 1. This is a region is a cluster 1. Here it's a cluster 1, and this is a cluster 4. It's a cluster 2, and it's a cluster 3. So we have done. So we validate actually. So we want to find whether this
Krishnan, humble request. Can you conclude, because our next speaker is ready? Can you conclude, please?
Yes, it's going to finish. Yeah, this is a cluster 2 and cluster 3, and this is a prognostic model showing the number of significant principal common analysis. Then this we also studied some heat map principal common analysis of PCS showing this interaction with this different Asian bamboos, and we found that this also. Then this also we did the population structure. Based on the population structure, we found 4 population structures, and this is the population structure. Yeah, you can see the population 1, population 2, population 3, population 4. Then this we found in the what population and which population, how many species are present. So this is the membership, the population. Then we also validated the population structure of these member species. Then we found that conclusion, each of the IREP primers had a unique differentiation, and this as a marker system offered a highly efficient and reproducible allele for studying inter-heterogeneous genetic diversity. Then AMAV also we found that the result suggested that the bamboo are not well evolutionarily diversified, although geographic precision could have occurred at a limited level. This study highlights the usability of IRP markers in determining the interspecies variability of Asian bamboo. And this paper we published in the Forest, it's a 2.5 impact factor. And we also published one more paper, bamboo, how the bamboo grows in the mosaic past growth. There's a tree physiology, one of the very good journals, 4 impact factor. And this is some of the, maybe next presentation, I will give a very interesting talk about the development of epigenetic mechanism of bamboo seedlings under hold. This is a complete epigenetic mechanism of the bamboo, mosaic bamboo. So now we are working on epigenetic modification of some of RNA and this plant translation, ribosome sequencing. This is our ongoing project. And we have also collaboration with the International Network for Bamboo and Rattan Species. So they have collaboration in India to increase the bamboo productivity, especially in the Northeast India. They visited Northeast India. They are establishing bamboo garden, bamboo training, all the workshops that they are establishing. This is the training, everything they are working. Also, I have been invited last year to develop the sustainable from various countries. Behalf of India, I participated. This is myself, and this is a different countries, especially from Italy, Indonesia, Myanmar, Singapore, Australia, USA. So we made concluded to create the awareness among the people to cultivate the bamboo, because the bamboo is one of the first species for the sustainable development. So with this, we are working. Maybe this is my current. I'm working with Nanjing. This is my current director. He is my postdoc supervisor, and I know he is my PhD supervisor. He has written many books, many biotechnology books. Now he is in the Palayangotte Trichy. He was a vice chancellor of the Madras University, vice chancellor of the Bharatiya University. He was a principal of the Loyola College, and he was more than 20 years of the director of Entomology Research Institute. And there is also the renowned professor like Mother Inasimuth, the Ananda Krishnan, T. Ananda Krishnan. He was such a inaugural director of this institution. After followed by he was the director of this institution. Now the another director is Maria Paikum. So with this, I thank you very much for giving me this golden opportunity to present my work. So thank you very much, sir. I am very happy to answer questions.
Dr. Ramakrishnan, actually, the next speaker is waiting for us. He's Professor Vasuki Belavadi from GKVK. Thank you very much for making our morning very beautiful by showing your good work on bamboo and its genetic diversity. We are on behalf of Department of Botany, Telangana University, and our participants. I wholeheartedly express my gratitude towards your presentation. Thank you very much. Belavadi sir, can you please switch on your camera? Sir, you can start sharing your screen, sir, actually.
Yeah, okay.
So he's our next speaker. He's our next speaker, Professor V.V. Bellavadi. Now I request our colleague, Professor M. Aruna ma'am, to kindly give his introduction. Aruna ma'am, over to you.
Very good morning. I deem it as a privilege to introduce Professor Vasuki V. Bellavadi, Sir, Emeritus Scientist from Department of Entomology, University of Agricultural Sciences, GKVK Campus, Bangalore. And his area of specialization is pollination ecology, taxonomy of solitary bees. And he has dealt entomology courses for BSc Agricultural Sciences, MSc Agricultural Sciences, and PhD program at UAS Bangalore. And he has handled research and operated 9 outside funded projects from DST, DBT, ICAR, Monsanto, and Coffee Board. And he has guided MSc students around 15 and also PhD students around 14. And he guided a Fulbright fellow from Emory University, USA, MS Lucy Anderson, on a research project on effects of sublethal agrochemical exposure on solitary bee reproductive fitness during 2014 to 2015. And coming to his publications, Sir has published more than 120 papers and accountable as popular articles around 12 and books around 2 and extension folders around 2 and manuals around 2. And he has delivered more than 100 guest lectures at various universities. And he has been conferred with many honors and awards. Few of the most popular ones and the most important ones are, he has been a visiting FAO fellow, University of Southampton, UK during 1995, and fellow of Royal Entomological Society, London since 1995, and R.L. Narasimhaswamy Memorial Award during 1998, and he has received H.S. Mehta Memorial Award in 2002, and consultant from FAO Global Pollination Project 2010 to 12, and is member of midterm evaluation team of Global Pollination Project for UNEP in 2012, and he has received ICAR Best Teacher Award in 2013, and is member of Research Advisory Committee ICAR NRC for Orchids and Sikkim, and is member of QRT to review AICRB on honeybees and pollinators. And with this, we do understand how great scientist he is. And with these few words, I welcome you, sir, and I invite you to give your lecture on the topic pollination and ecosystem service that is taken for granted. Seeing to the title itself, we do understand how interesting it is. And I request all the participants to pay attention and make a note of it. And thank you. Thank you for giving me this chance. Thank you, sir.
Thank you, Professor Aruna, for introducing me.
Thank you, sir.
After a wonderful talk by Dr. Ramakrishnan on DNA and genomes and all that, I think now I'll be speaking on some plants and insects. The topic of my presentation is pollination and ecosystem service, which is taken for granted. Pollination, as everybody knows, is an essential ecosystem service, and pollinators play an important role in supporting life on Earth. If you are looking at the entire biodiversity that is there at present on Earth, I would say we should be thanking pollinators. If you look at the history of life on Earth, flowering plants appeared around 160 million years ago. And around 135 million years ago, some insects like beetles discovered that pollen is edible. And that was the beginning of insect pollination, because when they started feeding on pollen, they started moving from flower to flower, accidentally dropping pollen grains on the stigma of other flowers. So that was the cross pollination. That was the beginning of cross-pollination. And even today, you will find that cycads are pollinated by beetles, and there are several species of plants which are pollinated by beetles also. Now, when plants started offering sugary nectar in addition to pollen, more insects started visiting flowers, I should say. With the arrival of bees around 110 million years ago, plants started competing for attracting pollinators, resulting in flowers of different colors, shapes, and sizes. So this resulted in a competition among plants to get pollinated or to be more attractive to the pollinators or the insect visitors. And there was a sudden increase and a significant increase in the number of plant species. And many people attribute this as a pollinator-mediated speciation in plants. In short, if we have so many species of plants around us, it is because of the competition that the plants had initially or even now for attracting pollinators, and that has resulted into so many species of plants. Why bother about pollinators is the next question. Pollinators are essential for reproduction of crops and wild plants. In short, nearly 87% or more than 87% of all plants, they depend directly or indirectly on flower visitors for their seed set. They are also vectors of genetic exchange. In a population, if you find a lot of variability, it is because of pollinators, because they are the ones which are actually carrying pollen from one flower to another flower, from one plant to another flower, and that is actually bringing about the variability in the plant population and ultimately leading to speciation also. And they also provide other ecosystem services. Like in the wild, supposing if there are several species of plants which are actually producing fruits on which many birds and animals are depending on for their own survival, it is because of the pollinators that are active on those species of plants. If the pollinators are not active or if they are not visiting those plants, they may not be able to set any fruits or seeds. So their propagation is affected and the life of many other organisms which are depending on those species of plants also get affected. So that is the ecosystem service that the pollinators are giving. Then of course they have a very high biodiversity value because, as I said, if we have so many plants around us, it is because of the pollinators that we have around us. And if the pollinators were not there, probably the diversity would have been very, very less in any given place. And the last point that I would like to make is the nutritional security of any country depends on pollinators. There are many species of plants which we cultivate for our nutritional benefits, like vegetables, fruits, pulses, and oil seeds. All these depend directly or indirectly on pollinators for their setting. And we might have achieved food security in the country, but for achieving nutritional security, we have to conserve pollinators. Now, one of the myths that we have amongst us is that plants bear flowers which become fruits. We see flowers around us in any crop or in any place where we go, we see a lot of flowers, and we assume that all these flowers are going to set fruits or set seeds. We just take pollination for granted because we do not try to look at what makes all these flowers into fruits or ultimately convert these flowers into fruits. There is a very famous statement by an economist, Pawan Sukhdev, who was an economist with the United Nations. In one of his deep reports, he says that not a single bee has ever sent you an invoice, and that's the part of the problem. Because most of what comes to us from nature is free, because it is not invoiced, because it is not priced, because it is not traded in markets, we tend to ignore it. So for whatever item that we are not paying, we take that it is free. We consider pollination as a free ecosystem service. This is the unfortunate thing. Now, the next question is, how many flowering plants are there? I said that there is high diversity of plants in the plant kingdom, but how many species of flowering plants are there in the world? According to a recent estimate, which was published in 2016, there are an estimated 295,000 species of flowering plants in the world. Some estimates say that there might be 369,000 species of flowering plants, but this is the most recent one. Now, 87.5% of the species of plants entirely or partially depend on flower visitors for successful seed set. Now, flower visitors in the sense that it is not necessarily only insects, there may be birds, there may be bats, there may be mammals which are involved in this, in the pollination. But insects are the most frequent, and among insects, it is the bees which are most frequent or most efficient pollinators of majority of the plants around. In India, we have 21,411 species of flowering plants, according to a report by ENVIS, Center for Floral Diversity 2018. And of these, 16,000 species require pollinating agents. So this is in short, what is the importance of pollinators when we are considering so many species of plants around us. Now, when we take into consideration the food crops, around the globe, we have about 115 species of plants which we use for food. And of this, 87 depend on insects for pollination. And according to Klein 2007, 35% of the global food production can be attributed to the service by pollinators. If you look at this figure from the economics point of view or the monetary point of view, you see that the annual economic value of pollination service worldwide is about 577 billion US dollars. And still, we take pollination for granted. There is a very interesting work by Basu et al., which was done about 10 years back in 2011, in which they classified all the crops that are cultivated in India as those crops which are dependent on pollinators, crops which are highly dependent on pollinators, and crops which are independent of pollinators. That means they do not require any pollination, like our cereals, rice, wheat, jowar, many of the millets, and all these, they do not require any pollinators. They are self-pollinated, or maybe to some extent wind-pollinated. Now, they found that in all those crops which are not dependent on pollinators or which are pollinator independent, over the years, there has been a steady increase in the productivity or the percent increase in yield over the previous years. Whereas this did not happen, or this was not seen with all those crops which depended on pollinators or which depended very highly on pollinators, or crops with high dependence on pollinators, or crops which are dependent on pollinators. They found that there is a plateau, or there is a stagnation in the yield after the 1980s. The increase in the yield with the pollinator independent crops is mainly because of the efforts by the plant breeders, agronomists, and all, who actually looked at the varieties which were giving higher yields. They selected high yielding varieties and then brought them under cultivation. And the agronomists helped in increasing the nutritional requirement of those plants and then tried to achieve the potentiality of the variety. Whereas the same thing was not seen with those crops which are highly dependent on pollinators or pollinator dependent, because the same breeders, the same agronomists were working on these crops also, but this is the problem. And very recently, it has been observed that all these crops which are dependent on pollinators, we are not, say, recognizing the service of the pollinators, or we are not doing anything towards conservation of these pollinators. So the conservation of pollinators might help, or increasing the pollinator populations might help in increasing the productivity of these crops also. So that is the thing which we have to look at. So are we ignoring pollinators? In fact, there was a very famous book, The Forgotten Pollinators by Bookman and Nobbin, published in 1996, in which they actually touched upon this point, that pollinators are being neglected in most of the cultivated and also in the wild areas. Now, this is the book which actually opened the eyes of policymakers the world over, including the Food and Agriculture Organization, wherein they brought in the Global Pollination Project to actually increase the knowledge of people about pollinators, including the policymakers and also scientists and the farmers. So this is a very important book that one should actually read. Now, when we say that, when we talk about pollinators, normally we say that the plants are pollinated by bees. Now, there are so many other pollinators also, like there are butterflies, there are moths, there are flies, there are beetles, and there are many species of birds, there are many species of bats, which are excellent pollinators, including apes and like lemurs, which are very important pollinators of some trees. Now, why we consider bees as major pollinators is the fact that bees actually are efficient pollinators. They have one great advantage over other pollinators because they can go back to the same plant repeatedly. So if a particular species of plant is flowering, the bees continue to visit the same plant or the same flowers so that the efficiency of pollination is more with bees. Now, when we talk about bees, we have yet another myth that when we think of bees, we think of only honeybees. Now, in the entire world, there are just over a dozen of honeybees, but there are something like 20,400 species of bees in the world. There are so many species, and an estimate says that there may be more than 25,000 species in the world. And in India, we have about 740 species of bees, including many solitary bees. Majority of them are solitary bees. They are not honeybees. They are not social bees, but they are all excellent pollinators of several crops, several species of plants. So we cannot ignore them. We cannot say that we worry only about honeybees when we are talking about pollination, but we also should see that other bees are also important. There may be several more species, as I said. Now I'll try to take one example of our own work on small cardamom, which is Elateria cardamomum, which is 100% dependent on bees for pollination. And if the bees are not there, this particular crop will not be able to set any seeds. So this is any capsules at all. So bees are very, very important. Honeybees are major pollinators of this, though there are other solitary bees which are also excellent pollinators of cardamom. Now, cardamom, as you know, is a commercial spice crop. It is also called as the queen of spices. It's cultivated in the Western Ghats of India. It is one of the major commercial crops of India, and it depends on bees for pollination and fruit set. Now, it is a zingiberraceous plant that is cultivated for its most valued capsules under the shade of trees. It is cultivated under the natural shade of trees. And if the trees are not there due to some reason, they plant shade trees and then cultivate cardamom. Now, in India, cardamom is cultivated over an area of about 69,200 hectares in southern states of India, mainly Kerala, Karnataka, and Tamil Nadu. Kerala has about 60 to 70% of the area, Karnataka about 30%, and Tamil Nadu about 10% of the area under cardamom. The annual production, I would say that it is a very important crop because the annual production of cardamom capsules is about 14,000 tons, of which 2,500 tons are exported, fetching about 250 crores of rupees annually to the exchequer of the country. So it is a very important crop which is cultivated for own consumption, internal consumption, and also for export. Now it grows in clumps of 25 to 30 pseudostems. So each one is a pseudostem. That means each year you get a different pseudostem. That's why it's called as a pseudostem. Each year you get a separate stem on which it bears at the base 2 or 3 panicles. So each year you find new panicles with flowers. Okay, so that's the reason over years a single clump may have many clumps, many pseudostems. So on an average, about 25 to 30 pseudostems will be there. And each new pseudostem will have 2 or 3 panicles with flowers. So the flowers are borne at the base of the plant, and the bee, if it has to visit, it has to come to the ground level and then circle around the plant visiting the flowers. And it is very easy to observe these bees also to work on the foraging behavior and all that. And a cardamom flower has about 23 millimeters long corolla tube, which actually holds the nectar. And when it is full, when a bee is not visiting, it holds nectar of about 4 microliters of nectar when it is full. And the flower also offers a lot of pollen to the pollinators, and it has an enlarged flabellum for pollinators to land. Now, these are the nectar guides. Okay, so the bee lands on this, and then it thrusts its mouth parts into this, and while doing so, its thorax and part of the head comes in contact with the stigma and also the anthers. So when it is going in, it may deposit pollen grains on the stigma, and then when it is coming out, it may carry some pollen outside. So this is how the pollination is affected. So when it is coming in, it brings the pollen from another plant or another flower, and then it deposits the pollen on the stigma, and the pollen, the anthers or the stamens below will have a lot of pollen, which it is going to carry when it is going back, when it removes its head and thorax out. So this is the thing. And these are the nectar guides which guide the bee to keep its where the nectar is. So that is how the bee lands and then takes nectar. Now coming to the floral biology of cardamom, the flowering is from April to November. Anthesis is in the early morning between 4:30 and 6:30 a.m. And anthers are hercogamous. That means the anthers are shorter than the position of the stigma. So the possibility of self-pollination is almost zero. There is absolutely no possibility of self-pollination, and the pollen grains are sticky and they stick to the body of the body hairs of the bee when the bee is visiting. And the pollen dehiscence is around 7:30 a.m. and pollen remains viable throughout the day. You see that the bees are active throughout the day, and the stigma receptivity is peak around 12 noon, and the flower longevity is a single day. That means the flower opens in the morning and it has to get pollinated on the same day. Every day when you visit a cardamom field, you'll find new flowers. So the flower, if it is not pollinated, it just withers off by the end of the day. Now, the major flower visitors are Apis cerana and Apis dorsata, the honeybees. And in addition to this, there are many species of solitary bees which visit the flowers. Now, interestingly, the percent fruit set in cardamom under natural conditions, we found it to be just around 30%. That means out of every 100 flowers that are produced, only about 30 or less than 30 are actually setting fruits. So this actually gave us a question whether there is a deficit of pollinators, whether the pollinator activity is low, pollinator number is low. And supposing if we improve the numbers of pollinators or the bees, whether we can increase the fruit set or yield in cardamom. So what we did was we just made some calculations based on our foraging behavior studies. We had found that the number of flowers per clump that is available every day is about 20 flowers per day is at every clump. So if the number of clumps or the plant population in a hectare is about 3,000, so that means to say the number of flowers every day that is available to the bees is about 60,000. 60,000 flowers are available every day. Now, the number of visits that a flower receives is about 20. You know that the flower has multi-ovuled. Okay, the number of ovules is about 27. So if majority of the ovules have to be fertilized, it requires more number of visits, more number of pollen grains to be deposited on the stigma. So it receives at least about 20 flowers. So we have taken the minimum number of visits that a flower receives for setting. And a flower visited by a single bee in its bout, when it comes to the cardamom field, is about 130. This we have found by tracking individually marked bees about how many flowers it visits, and we have also measured the quantity of nectar that the individual bee is taking. If the individual bee gets only about 0.3 microliters of nectar, and the stomach capacity of the bee is about 40 microliters, and so to fill its stomach, it has to visit about 130 flowers. So the number of visits required per day is about 1.2 million, or 12 lakh visits are required. So we calculated that the number of foragers required per hectare would be around 9,000, which comes to approximately about 2 colonies of bees. So we did a field experiment in which we actually placed 2 colonies. We had 3 different plots separated. One is where the plot is completely caged, where no bees are allowed, and an open pollinated plot where the feral bees or the naturally available bee colonies or bees are visiting. And in another plot, we introduced 2 colonies, and we found that you can actually almost double the pot set if you say keep 2 colonies per hectare. So this we have actually given as a say passed it on to the farmers, and then we have done a lot of field experiments also, field trials also with this. Now, honeybees, we have found that are essential for fruit set in cardamom. And by introducing 2 colonies of bees per hectare, we can almost double the yield of cardamom. This was the conclusion of that work. Now, another important aspect is it is also necessary to put a value for the pollination service. So when we say that the pollinators are important, the policymakers or people in general will understand what is the actual benefit that we are getting. I already told that we have the economic value of the pollination service is about $577 billion US. But when it comes to a single crop, whether we can measure this. So we have done this with cardamom with what is called as the economic value of pollination service, EVP, with a formula that pollinator dependency index. You can calculate the pollinator dependency index for each individual crop by how important that crop is. There is a method available for this. And if it is 100% dependent on pollinator or highly dependent on pollinators, you take it as 0.95. And if it is on an average, if it is benefited by the pollinator visitation, it's taken as 0.65. And if it is also highly self-pollinated and benefits by pollinator visitation, it is taken as 0.35. And if there is no pollinators, or occasionally if the pollinators are visiting, it is taken as 0.05. And if it is self-pollinated, it is 0. So that is a thing it has been worked out. So it comes to pollinator dependency index varies from 0.05 to 0.95. Now, PR is the productivity of the crop. That means the production per unit area. And the total area under the crop is A. So in any given state or any given country, the total area of the crop is taken into consideration. The average productivity is taken into consideration, and the market value of the product per unit is taken into consideration. For market value may be value per ton or value per kg. The market value is taken, or in some cases, you can take even the farm gate value, farm gate price of the product. Now, if you put this formula to the cardamom production in the country, you see that the PDI is 0.95 because it is very highly dependent on pollinators. The productivity, the average productivity in the country is 187 kg per hectare. And the area under cardamom, the exact area is 69,132 hectares in 2019. And the value is 1,000 rupees per kg at that time. So the EVP, the economic value of pollination service to cardamom, it comes to 1,228 crores of rupees. So should we not conserve pollinators or improve pollination in cardamom? So if you look at, again, the annual economic value of pollination service worldwide as 577 billion US dollars, it comes to 42 lakh crore rupees. So, and it is not just the money, but the actual value lies in the total biodiversity that is conserved by the pollinators when you conserve other pollinators and protect them, bees and other pollinators, and protect them. By conserving pollinators, let us enhance our nutritional security and also save nature. So at the end, I would like to change the title of my topic as pollination and ecosystem surveys that is not to be taken for granted. And if we take it for granted, probably we will have markets like this without any produce. And supposing if you protect the pollinators, maybe we will have a market like this where you have everything, fruits, vegetables, everything, pulses, everything available for us. And this is my team of students. Thank you very much for giving me an opportunity for me to speak about my favorite subject. Thank you very much.
Thank you so much, sir. Thank you very much for giving an enlightening talk on this issue, that is pollination and the role of insects. On behalf of Department of Botany, it is my deep sense of gratitude towards your such a beautiful and wonderful lectures. And we expect same kind of cooperation in future also for many more events. Thank you so much and have a
Thank you,
great.
sir.
Thank you so much for joining us. Thank you so much.
Yeah.
So now, with this, we have come to an end of technical session 1, and now the technical session 2 begins. I hope Indira Madam is there in the meeting. Indira Madam. Indira Madam. Now, technical session 2 begins with talk by Indira Madam from New Zealand. Indira Madam, over to you. You can start your screen sharing and you can give your talk. Ma'am, your voice is not audible.
Are you able to?
Yes, yes.
Good evening. Thank you for the wonderful opportunity. It was really amazing listening to Dr. Ramakrishnan and Dr. Balwani. There were so many insights that we have to keep learning, and there are so many wonders of the world that nature keeps showing us through different eyes. So it was an amazing session listening to both of them. And I have chosen a general perspective being a teacher. So I thought, and I have been a teacher in India, and also I've grown from primary teaching to tertiary teaching, as well as I migrated to New Zealand, and then did this repetition of probably the same thing from early childhood to tertiary teaching. So I thought I would be teaching, sharing my experiences as a science perspective, the importance of science in a daily, everyday living and learning, but mine would be more related to general perspective, or from a student's perspective, from the way the teachers from India as well as in New Zealand, that I have learned and I have seen in my students, the enthusiasm that they have shown towards this subject. So, so the role of science in everyday living and learning. So science plays a very important aspect. Science is, it gives us the opportunity to explore within ourselves how our body mechanisms work, and around us, the environment. And so science does play a very important, and to briefly sum it up, science is the pursuit and application of knowledge and understanding of the natural and social world following the systematic methodology based on an evidence. So it is factually, it's based on facts. So we all know that scientists, when we do experiments, we take time to set up the experiment, follow the procedure, make observations, record the data, and then analyze and try to see if it aligns with our hypothesis and what results we found, and how we have performed, and what we could expect from that experiment, and how we share it findings with our fellow students, colleagues, or writing reports, and try to find answers to the problems that we are trying to find. So science does play an important role in everybody's life. So I think our day starts with science. So the next slide. So some of the examples that I think where our day starts with science, to be honest, so right from the toothpaste to the dessert that we end of the day. So everything is made of some sort of chemical that comes, the nature has provided us. So we find science being used, the knowledge of science being used in different products or the tools that we use from the start of our day to the finish of our day, and through not only in those two aspects, but all through the day as well. So we use scientific methods, scientific advancement of the tools that we try to use, make these gadgets to make our lives more convenient, and we try to simplify things. For example, the best example would be a touch screen, where we think by using gadgets, we have become more movement restricted, I guess. So if I can put it in, so it is imperative to understand that science helps us in our advancement, and we try to seek solutions to all the existing problems, trying to achieve or simplify human lives as such. So plants and animals do play an important role, but the importance from the place that where I am looking at, coming from a recent, I was my last assignment was with the nursing students. So the students were more, of course, with the COVID things and happening, more of health and hygiene was the priority all over the world. So that was the same across the board. And for the students coming from the primary student, if I was dealing with a primary class student, that would be a different approach. They use the sanitizer and the health and the hygiene, but the perception of understanding of the tools and the importance of science and the reason why we do experiments to find out the facts, and how we try to collect the data, and how we try to utilize that information to better the products or the living conditions that we are living in. So the knowledge varies at different age levels, and the criteria depends upon the student or the community of the school community also, because each community has a different set of expectations. And in New Zealand, the only difference I would say in India, we had the same curriculum across the nation. But whereas in New Zealand, I would say that it is confined to the small school community, and they design their own study and their own choices of learning. So as a result, the students have more opportunity to open up and try to pursue experiments or research or projects in a it's more of a student oriented or a student directed learning, in other words. So that way they have more opportunities to do that. And in that process, now science has become a more integrated curriculum. So science cannot be taught as a separate subject. It has become a merger of curriculum with this maths, English, arts and technology to make it into a STEM project. So, and some schools use it as a STEAM, inclusion of arts. And so it has become a mix. This undoubtedly has given opportunities for people who enjoyed science in spite of the difficult terminology. Some of them find it, the terminology to be a hindrance for some students because they find it really hard to understand it. But if they get into like-minded, or if they're able to be helped with to break it down and be able to see the meaning for the word. For example, in most of the science words, you can see mon as a bad and benign is good, and thing micro is small and macro. So all this terminology, if you can break them down to the students of primary and second, so it can be simplified. My plea would only be to make it more simplified so that it should not be a stumbling block for students to pursue science. So that would be my request and plea for the parents and the students or my colleagues to make it as simplified as possible for them, for the students to be able to enjoy science because it's more hands-on, and they can learn by doing. They don't have to mm, by heart the theories, and they can actually believing the whatever the problem that they are looking at, they can do it by hands-on approach using things that are available to them in and around within their community and within their school facility or within the parents. And with the STEM projects, there are so many opportunities to be pursuing science at every level, within the school community, in the school and outside, outdoor activities, extracurricular activities, and some school projects. And so I, as a teacher from growing from primary, early childhood, as the secondary and tertiary, I find students, I also try to go across the sector sometimes. So which also gives me an opportunity to understand what the current needs are. And also being in New Zealand, I had the opportunity to have personal professional development in the required area. So that also depends on the school community and individual school needs. So they send you on professional developments and you can learn. And as a teacher, you can actually facilitate more. Teaching is more of child ownership these days. So it has changed. There is no more teacher centeredness. The teacher only facilitates and actually, in fact, learns alongside the student. So this opportunity, or this change in the trend of teaching environment actually plays a significant role on the student by giving them the opportunity to learn, uh, be proactive. And if it's a very proactive student, the teacher can actually facilitate and help them to achieve better. For a gifted student, for an example, so giving them the projects to especially boys with year 3-4 classes, they are more into making cars and things, and trying to find ways and means of recycling. So these are the different projects that whatever the student and the community as the school can provide, this gives them opportunity, because there is no fixed curriculum as such. So you can actually there's an overarching learning outcome, and but from that, the student and the teacher can negotiate and find out various means of experiments and projects to build through. And as a result, it gives opportunities for the individual to excel and further their love for science as such. So understanding of ecosystem, having an outdoor visits and making understanding the life cycle of a butterfly, for example, it's able to see all the stages of the butterfly within the classroom is a unique opportunity, as well an exam for the child to see and actually understand and see how the life cycle happens. And it from there, they can apply the knowledge to different animals and see different insects, and their observational skills and inquiry skills actually take on further and lead them to further investigation and research aspects. And that also helps them to sometimes it gives them, there are students who can work individually, there are opportunities for them to work within groups. So that also builds their personality and communication skills and overall developmental skills, to see how their interaction and how they could actually enjoy doing that project, and what would be their next steps and what would they like to achieve from there onwards. So the recent one would be where there was a high school students were working on a project called with the creating a fuel efficient car. So they were understanding the drawbacks of the carbon emissions. They have done a lot of research on different countries, how the cars have been polluting the atmosphere, and what are the impacts on the atmosphere, and how it has been also adding to the revenue of each of the countries. And then now they have come up with a different, after the research, they have come up with the solar panels, the windmills, and they have made models of all different kinds of things. So this gave them an opportunity to discuss and have an invite and also have conversations with expert scientists and engineers to have that conversation at 10 years and 15 years is really a unique opportunity for one to open up and to able to see the bigger picture in their life and to see what that science and things can hold and what their life or the career choices could be based on that. So science, teaching science requires a lot of preparation. So with the students, with the negotiation with the lab attendant, the technician, to be able to organize and to be able to see what the student needs are and to analyze what is possible within the budget of the school, and to be able to rationalize and also able to convince your departmental colleagues and try to give these students that opportunity to explore further, or give them an opportunity to take and see them, the animal that they want to visit, and see how its life cycle and how it actually, what its ecosystem is, and how, what are its food habits, what are its predators, and you can take them to different places where they, so this all involves a lot of organization and understanding of the child's interests. And so in order to create a plan or a program to cater to the needs of the student is sometimes a tumultuous task for the teacher, because you have different 30 students in a class and everybody has got at least you will be getting a couple of 5 different challenging problem or projects where you will have to find, or you will have to extend yourself as a teacher to make it happen for them. Even though, so those are the things that science actually, in the end, it is quite rewarding after they fulfill the experiment, when they feel satisfied, when they come up with joy and they with the excitement that they have, their results are published and how they, what their findings are, and they're able to share their findings. So that would give the rewarding moment for any teacher. So science has been it has been a live subject and everybody appreciates it, but there is also what is I personally find as a science teacher is we are totally science dependent. We right from the day and from dawn to dusk, you are always related with things that the appreciation of nature, how the seasons change, how the plants grow, the things around you, and the food that you eat comes from the plants, and how it can change and how it, you know, inspires individuals for the taste, the palate, and what are the things when you're when a properly, what happens when the person is overnourished. So all the problems that the society is currently facing world over. So you can discuss all those events. The students come up with different plans. They come up with a dietary plan for the obesities. What is the common problem? So obesity was discussed as a common societal problem. So they came up with a different plan for the teenagers, different plan for the early childhood siblings that they had, and they had come up with a plan for their old grandparents. So what are the things that they could be? And similarly, they came up with different kinds of exercises, physical needs. The science is evolved into so many different branches of physical science. So sports science, health and medical science. So they have got different ways of approaching writing letters, talking to the DHB, talking to the doctors, talking to the inviting the nurses and the specialists to come and talk to them about rheumatism and things like that. So that was a common problem in one of the projects one of my students was doing. So they had invited a doctor, and so they were able to discuss the symptoms. They were able to create a booklet. So that was their final project, how they created the care for a rheumatic child who suffers from rheumatism, and what are the care and how can in order to provide a holistic care, the role of his siblings and role of the parents and the neighbors and his friends, what are their roles to be played. So everything comes into place as a holistic aspect. So all this came as a part of the health project that the students were doing. So, and then they have also discovered what are the careers that they could propose. So what are the because this world is changing, and the jobs that we had 15 or 20 years ago are not the same today, and the world is with the changing scenario with the COVID and everything. So people have realized that since some families have lost the jobs in air hostess and things like that. So people have realized that the job in healthcare is more reliable, and more science-oriented jobs are more stable. And that is what the present perception of amongst the students is, that anything to do with science, you will be able to sustain it, and that will lead to a future. And that is what the present trend here is. And I think it's the same with healthcare, and people are going into more of caregiving services, personal cares, and transport services. So this is how the knowledge of science has helped them also understand the things have changed, the need for the learning of science also helps them. It's though it seems to be the word terminology seems to be putting off some people, but yet if you are able to break it down and help them understand that, it gives them the confidence to learn and get over their fears and stigmas. So once the student gets over the stigmas, then they are able to see that the science information is there. We rely on the facts and the factual information. They can do it for themselves. They can see repeat the experiment. They have a systematic approach towards doing it. They follow the methodical approach, and they are able to hypothesize the experiment before they do it, say what could happen and what are the things that might. So checking the understanding and beliefs, and they also have the opportunity of interaction with the different professors and experts in the community. So that gives them a different and a deeper understanding, rather than just relying on superficial knowledge. So that helps them to unfurl the deeper concepts of the science, and they also able to see the different picture, getting the knowledge from the expert point of view. So this also makes them feel connected, and they can see the real world in problems, and they can actually analyze and see them more connected and relevant to their perception. So what they are actually facing or what they see in the community. So it makes learning more relevant when they can actually see and make sense of it. And it is more, when learning is more meaningful to them. So learning makes more sense, and that actually gives them the ownership. And that's when they come up and say, that's the project, I want to change the project a little bit, or they want to give it and they want to change their adapt, make a little bit change or adapt into a different. So these are the things that give them student or the learner the ability to pursue and interact with the different people in the society, to build relationships, to build their own network, and also to understand the society, how it goes, and the different aspects of science, actually how to look after the environment. Why is a plant need? Why do we need to take, and why do we need to prune it, for example? So all these things, it's a part of taking care of the plant. So how do we need to get rid of the debris, and why we need to do compost? So these aspects are embedded through them right from the early childhood stage, where their rubbish is sorted in the kitchen or whatever they use it. It's supposed to be going into the recycle. It's supposed to be going into the compost, where and the food waste. So they are able to classify, and they're able to look after their own personal garden and the personal environment, whether it's a classroom or it's a home environment, the child is already taught the scientific aspects. So they know that the food and this waste goes into the worm bin, where the worm bins can be looked after, and they are going to go check with this. They do have a help and support for so they ask for the gardener or the specialist to help them to help with the environmental project that they are doing, to look after the compost, to look after the garden in the pond and in the school, to nurture the trees and look after the aviary. And so the students take ownership. So it is their project. So they feel part of it. They feel proud, and they take responsibility to look after the project during their tenure and during their during that year of that classroom. And unless they move to a different, so they can extend on their project when they go to the next class, or they actually can try to build on it, or they can change the project, or they totally choose a different project. So the next class year gives them the opportunity to do both to suit the needs of the student. So student as the center of the learner, the systems. So he or she chooses the criteria, what scientific projects they want to do, and what kind of, whether it is to do with plants, to do with horticulture, or is whether if they want to do with the energy efficiency, or so depending on the need, what the child thinks it's a health problem, or so the child gets the opportunity to do that. So they all, the teachers or the facilitator role only is there to support and to build in that curiosity to see what they are doing, and try to be supportive of them and provide opportunities to help them to excel in the project that they are doing. So that is how science has become an important aspect, where it can doesn't have to be taught in isolation. It has to be integrated so that it can be taught well with maths, taught alongside in literature or in literacy English, and it also can be taught alongside of technology. So all the innovation comes through science, and then so that is how they can see, understand the different aspects of it. Even for the students are taught home science, or they're called they do cooking, where food science, where they actually understand the level of what is the quantity size, what is the slice of bread look like, for how is it made, and how many calories it has got, what is the process it undergoes, what does fermentation mean. So this learning of the making of the bread, or making as a toast or as a breakfast for himself, the child will learn all these concepts. If it's a year 1 and 2 class, we just try to give them the learning of the toast would be a bit starting point. But whereas if it's a down the road, if it's a middle school, elementary school child, then the child will be doing about atoms and compounds, and how the bonds happen, and what happens when the bread is heated, how does heat impact the bread. And it's a bit more processing and more knowledge and more analyzing, and the child or the student will be able to further investigate different aspects of temperature, heat, effect of heat, food, the interactions between molecules, what happens when the dough is added to water, and what happens when it is added to egg. So they try to see different types of reactions, and they try to analyze how many calories each gram has, and that is how they try to build their own menu. The list, the project would likely to end in creating a menu plan for them for the week, where their nutrition and is whether it's balanced, or whether they can create a be able to analyze what they should be eating, what is healthy food, and what are grains, and what are veggies, what are pulses, to be able to categorize food into different sections of fats, oils, proteins, minerals. So these all these aspects give them opportunities to be more realistic, be more authentic, and take projects from their own learning, own life, day-to-day life, and then they try to create projects, and then they try to create and share presentations and create their knowledge with other classmates, and they even share it with the fellow community of schools, community of learners. So science, learning of science has also opened, it's not only about, it has increased the knowledge about themselves in particular, how their body functions, why is it the arms, or why did they run fast, why was the, so they would be able to analyze the physical aspects of it, the physiological aspects of it. They would be understanding from, one can certainly see the growth of a child from basic science to going into deeper and deeper levels as the age increases, and their personal, depending also on their personal interest and where they are more likely to be proactive, when there is more support from the parents as well as, and also from the teachers. And those struggling students will also succeed in science, especially because it's a hands-on thing, and they don't have to be fearful of falling behind, like in maths, or which is a hard subject, or calculations and things like that, because science is more about doing and hands-on. So they know the process, they can make, the steps are written down, and they break it down into smaller steps for the child who is struggling, so that helps them to understand better. So science is such a subject which can be taught across all sections and all abilities of children. They all do enjoy it thoroughly well. They enjoy the nature, they enjoy the greenery, they enjoy the aesthetic aspect of it. So I think it is a responsibility of a teacher as an adult to help every child understand the appreciate the nature around us, that we are responsible for looking after it, and also for being able to use aspects of it. We are responsible to give it back to it. So we have to take care of a plant, for example. If you look at the nature, we take fruit of the plant, and then we need to look after it, give it nutrition, give it compost and other things for it to grow well, healthy, and keep looking after it. And so giving all these aspects, and it helps the child to understand the major impact, the major or the deeper concepts of the environment, and how we are all interrelated. Unless we don't help and look after each other, it is not possible that we all can live happily. So that's the whānau, or the family relationships, like it's a strong bond between you and your nature. So everybody has a responsibility towards nature to look after it, and then help. As a teacher, it also helps us to help them become a world citizen, where they can understand every geographic country. Geography also plays a part, where different climates come into place, where different what animals live in different parts, and climates, and how these climatic changes and things can impact their food habits, their way of dressing and things. So that also helps them to understand each other better, because cultures come into place at that point. So the students have the deeper their scientific knowledge, this appreciation for life and appreciation for themselves also increases, actually. And this science as such is an amazing subject, not only for being a science teacher, but also as a mother, and everybody has an interaction with the environment around us, whether it's plant, animal, and the human beings around us. We all owe it to ourselves that we understand ourselves first, and then also the environment around us. So for the students to understand these concepts, it is whether it is driven by the ministry or driven by the government, or the curriculum is driven by the student, or the choices made by the projects, the teacher still helps, it is imperative that we all help each other and to appreciate nature, and also to look after nature. So then we will be able to sustain ourselves and also keep it going for the future generations. So that is our responsibility as a world citizen to look after. So the constraints could be the curriculum driven sometimes can be very difficult to extend the students if it is curriculum driven. So
Are you listening to me?
if it's an overarching, and if the teacher has the flexibility to provide more, or can give them diverse opportunity to think beyond and come up with their own ideas to research and things, so it would be able to facilitate the students' ownership gains. The student agency is promoted, and as a result the students become more independent, and they also look at things in a more rational, and because of doing experiments constantly and trying to analyze their projects and following the scientific method to do the experiments, and they also develop a keen sense of appreciation. They also do things in a more methodical manner, and they also have a developed understanding of how things can happen and why things happen for a reason. Their understanding is quite phenomenal in those things. The constraints that are normally in order to facilitate science are generally adaptability comes at whenever the curriculum changes or there's a new thing coming out in the markets, the children are more into it. So then adapting to the latest things would be a difficult thing. So for example, we cannot create robots and drones at school. So those little things, the children may come up with some fancy idea, we want to create this. So you could, those are the little things that adaptability, because funding and things can become an issue. So those things will be difficult for any institute to facilitate such things. And having staff trained for facilitating them also is a thing. Not all will be inclined with all areas of the science, because science is so broad and you can have interest in different aspects of science. And to facilitate for 30 different students, it would be a difficult thing for the teacher, unless they have the they themselves have the keen love for science and trying to explore, and they themselves are keen to learn and to try alongside their students. And then to break down the resource availability, because it depends on the budget and the school's resources, so that not and then the resistance to change could be an attitude with some old some personnel who will not be able to give adaptation students much opportunity to trial. So that becomes a difficult aspect for the child to understand that science is beautiful. When we say it, we must be able to give that opportunity. And if we try to put constraints saying that it's costly or it is unnecessary at this stage of, then the student will not be able to take ownership or believe in us, to be honest, if we do not if we do not walk our talk. So as a teacher, it is imperative to show that science is science.
Indira ma'am, can you conclude, because next speaker is ready already?
Okay. These are some links that I have to explore. I have put over there, and then the good resources would be to use people from everybody is knowledgeable. So knowledge comes from many spheres. We can so learning happens in mutual space. It's learning, it doesn't start and end on a fixed date. So we can keep learning from each other. So that's how we can learn. And then So from my personal beliefs, that science is an amazing discipline which relies on facts, observations, and follows methods, process, and values critiques also. It builds on keeps and helps us to keep moving forward. It finds solutions to all problems, and we are trying to keep on trying and excelling in things that we do. Science happen every day and everywhere. So science knowledge is happening for everyone, and understanding. It starts with understanding oneself, their own body, and the world around them. Science is an inherent understanding of one's own self, and then science is everywhere. So the knowledge of science is very imperative, and it helps everybody to understand themselves first and the environment, and we owe it to ourselves to excel in science. Thank you all.
Thank you very much, ma'am, for giving such a spacious time for us and good knowledge what you have shared with us. I express my heartfelt thanks on behalf of Department of Botany, Telangana University, for accepting such accepting the invitation. Now over to Vidyavardhini madam. Please, can you introduce our next speaker? Daniel sir, can you start sharing your screen?
Yeah, sure.
Good morning, everyone. It's a privilege to introduce Professor Maman Daniel. He's a professor and ex-head Department of Botany and ex-dean Faculty of Science, MS University of Baroda. And at present, he is the managing director, Dr. Daniel's Laboratories at Baroda. He has 39 years of teaching experience and 49 years of research experience, and he has supervised about 40 MSc projects and 17 PhD programs and 5 major projects. He has completed MSc in the year 1971 and PhD in 76, and he's working from 1979 onwards until 1997, where he worked as a professor. His field of specialization is taxonomy, phytochemistry, phytoalexins, and medicinal plants. He has published around 170 papers and 18 books. And among the 18 books, 5 are international publications. And one of the few of the popular books include Healing Herbs for Ease, a book aimed at plants useful for beauty and health, and an Amla, an indigenous tree with enormous potentialities. He has published around 167 research articles, and he has guided around 17 PhD students. And he has other academic achievements, like he was a chairman faculty board, Faculty of Science, MS University in the year 2009, member faculty board, Faculty of Science since 1991 till 2011. And he was the chairman for 5 years and member board of studies of botany since 1985 to 2011. And his present assignments include member research advisory monitoring committee, RAMC, for Botanical Survey of India, appointed by the Ministry of Environment and Forestry, Government of India. He's a member advisory board for Mentor Biological Sciences, Navacharana University, Baroda. He's a member board of studies in botany, Nagpur University. He's a visiting professor, Karul University, Baroda. And he's a visiting professor for Government Ayurvedic College, Baroda. And he has completed a few major projects like allelopathy and phytochemical studies on some trees introduced in social forestry, funded by UGC, and an in-depth study and documentation of floristic components of Gujarat for trends in speciation, utility, and bioprospecting, funded by UGC. He's a coordinator for the DST PIS program, Department of Botany, MSU, and he's a medicinal plant garden for conservation, preservation, and development, funded by National Medical Plant Board, New Delhi, from Jan 2008 to March 2011. And he also was involved in a project ex situ conservation effect through strengthening of existing botanical gardens through the implementation of ESM plan for CAD phase 1. And he has received a few awards like Lifetime Achievement Award in Life Sciences by Indian Society of Solid State Chemists and Allied Branches at International Conference on Futuristic Materials in Forensic Science and Life Sciences, Department of Chemistry in Nagpur. And he even received Marottama Manava Seva Award 2015, the highest recognition from the Syrian Christian Marottama Church, Kerala, in the year 2015. And with this, I would welcome Professor Daniel, to give his message, his presentation. Thank you. Over to you, sir.
Thank you, Vidya madam. Now, am I audible? Hello?
Sir, you're audible. Yes, you're audible, sir.
And the screen sharing is fine?
Yes, it is visible.
Now, can you expand it, the screen sharing?
Sir, on this, there is a last where you can see the slideshow. Not that one, sir. This here in the top also it is there, sir. Here, sir, top in that you have slideshow like that. You can click that. Not that one, sir. Not that one. Sir, slideshow. You come far. Or down also you can come, sir. Down also you can come. This percentage is there, no? After that percentage, there is one screen.
Yeah.
Not that one, sir. This one.
I can't see you.
Sir, down you can see, sir. Down there is a. This one is there, sir. Yeah, go up, sir. There itself. Go up. Just go up, sir. Yeah, that one. Click it, sir. Click it. Yeah, now it is okay, sir. Now it is okay.
Now, good morning, all of you. I think it's already late by about 20 minutes, and I have a large number of slides. I don't know how I'll finish within 30 minutes, but I'll try to complete whatever I wanted to tell you. And this presentation is with Dr. Ahmed, and you can share it. You can get it from him. Now, botany is a least preferred subject. They're the last choice of every student in science. Now, but there are so many business opportunities in the field of plants, especially in medicinal plants. See, there are so many others are there, which I'll just touch at the end, like natural dyes, biopesticide, biofuel, biofertilizer, etc. But today I'll be concentrating on the business opportunities in the field of medicinal plant. You just see the slide, the Ayurveda market, there's a herbal market. I will be explaining what is Ayurveda and what is herbal. It's Indian rupees, about 300 billion in 2018. And within 6 years, it will double more than that. And it will come to 710 billion. And the number of manufacturing units are 9,000 linked with ADMA. Then there are more than 9,000 who are using loan licenses. And there are more than double the number having third-party manufacturing. So the total number of manufacturing units may be about 40,000. So the needs of medicinal plant is very large, very large. And therefore, I'll just tell you, what is the next? Now, there are three options. Say, you can, being a botanist, you can supply the raw material. See, to supply original raw material, the correct raw material to 40,000 industries will be a huge one. There is one scope. Second is you can manufacture extracts. You need not manufacture a medicine, but you can manufacture extracts and supply to the industries. Industries prefer extracts because transportation charges will be much lesser, and then storage will be quicker, and then they can just mix the extract and make a medicine. And third is manufacturing medicines. I'll be mainly pointing to the manufacturing of medicines. But before that, I'll come to what is the raw material supply. Now, 80% of the raw material available in the market of herbal medicine are adulterated, are wrong. mainly adulterated and many times wrong raw materials are used. They do not get botanically correct raw materials. So a botanist has the advantage of identifying the botanically correct plants. And when you start collect only healthy plants. No diseased or insect bitten plants are to be collected. Then if you collect it by flowering or fruiting stage, that will be ideal. Don't collect the seedlings. And if you want only leaves, for example, I use Tylophora asthmatica, and we need mature leaves. So the leaves are to be collected from 5th node onwards. Now, all these materials should be tried in the shade. Or if you have facility, you can try them in an oven at 60 degrees centigrade, not more than 60 degrees. Then try it and powder it and pack with disinfectants. And then you certify that. You see, being a botanist, you can certify that this is the original material. And the original material will have a lot of value in that. And many people, if you can establish, many people will go for this. Now, if you want to extract plants and sell plant extracts, 2-3 points you will have to keep in mind is that you will have to extract them, boil the plant material in deionized water. Don't use the tubular water or the piped water because they contain salts and all these salts will remain within your extracts. So boiling in deionized water is the first thing. Then the extract is to be filtered and stored. And then the residue has to be extracted 2 more times with fresh water. Because normally people boil the plant material for about 2 hours, means 10 times plant water for 2 hours, then filter and concentrate the extract. This has a lot of problems because in the first extract, first 2-hour extract, only the high soluble compounds will come out. And then the extract become concentrated. So no more compound will get soluble in that, and the extract becomes saturated with certain other compounds. And you know that a saturated solution cannot extract more compound. So we have seen that it will first extract maybe about 60% of the compound will come out, then filter it, take the residue, extract it with again the same amount of water. That means if you take 50 kilos of material, extract them with 500 liters of ionized water. That is the first extract. Again, the residue, another 500 liters of deionized water. Then in the second extract, you get 25% more. That means the first extract 60%, second is 25, so 85%. Then again filter it and the residue is to be extracted with another 500 liters of water for about 2 hours. That will bring about 10% of the remaining compound. That means totally it will be about 90 to 95% of the compound will come. More than that, you will not get it. Otherwise, you will end up with 50 or 60% of the compound. And the yield also, if you take 50 kilos of plant material, if you take only first extract, you may get about 5 kilos of extract. But if you have 3 extracts, then you may end up with 8 to 10 kilos of extract. So that is more profiting. And these 3 extracts are to be pooled and concentrated. And the concentration, maybe if you have money, you can go for a vacuum dryer. Just like, they do it with milk and milk products. And then preservatives are to be added. And if you want to extract the insoluble, the compound insoluble in water, like oil or resins or lipids or steroids, you can extract them with isopropyl alcohol. Now, for that, you need a very good distillation set and expert. I know a group in Trivandrum. They are all PhDs from that tropical botanical garden. They are pooling 10 lakhs each and they have a capital of about 50 lakhs. They are going for these extracts with isopropyl alcohol and other compounds. So you can, if you can have a group and you can contribute some money, or you can go to any other industry which has the facility, and then you will have to give a certificate of the constituents. And then third and the easy form is manufacturing of Ayurvedic or herbal medicines. Now, Ayurvedic medicines are different from herbal medicines. In Ayurvedic medicine, you can use only those plants listed in old books, Charak Samhita, Ashtanga Hridaya, Aryabhishek, etc. You cannot add any new plant. And the methods of preparation are also, you will have to follow what is explained in that book. Then many a times polyherbal formulation containing 30 or more plants are common. See, for example, your Chavanprash contain 25 to 30 plants. And then you can add minerals as bhasma. And there is no concept of active principle. And no new plants, Indian or foreign, can be added, because I wanted to add stevia to one of my product and I couldn't get license. Now the new scientific methods of extraction and processing are discouraged. And there are a lot of restrictions on the food, if you go for an Ayurvedic medicine. Then contrast to that, in herbal medicine, you can use any plant provided they are discovered on bioprospecting, ethnobotany, or pharmacological or phytochemical screening. Now, there should be clinical evidence for that. So they are evidence-based pharmaceutical drugs that is common in all Western countries, especially Europe, USA, China, Korea, etc. And these are also based on herbal medicines, based on materia medica of Dioscorides. Now here, you will have to select the plant with a large amount of clinical researches. So you can amass all the available literature on that, then select your plant, then select the extraction method, and you can just concentrate your extract so that, higher percentage of active compounds are there, and you can enrich the active principle, and you can add antioxidants, antimicrobials, biocatalysts, immunomodulators, and there are no restrictions of food. And then if you want to manufacture a herbal or Ayurvedic medicine, say in India, we have only Ayurvedic medicines are available. Means you get a license for manufacturing Ayurvedic medicines, and no provision is there for herbal medicine. People are thinking in that line. Even the government machinery also is trying to route that way. So there are three ways. You can start your own manufacturing unit. If you have good amount of money, you will just see later. Or you can work with loan license, or you can go for third-party manufacturing. If you want to start your own manufacturing unit, it's a very laborious process. You need 1,200 square feet built-up area, 7 rooms, machinery, and laborers, and you need a number of equipments also. But most important is that you need an Ayurvedic doctor or an M.Pharm or B.Pharm in Ayurveda. Now, based on them only, they will give you license, and the area should be there. And there are many criteria are to be fulfilled. You can also start with a Vaid or Hakim or a pharmacist, and then you should have quality control laboratories, and number of things are there. Then once you have all these things, you can apply for manufacturing license number, apply for GST number. Then you will have to give a formulation, and that formulation, that is which product you want to approve. The name has to be approved. Then the components and the ratio are to be approved. And then you can send it to the ministry for license. And then you can start manufacturing. And then the problem is selling your products. And then manufacturing license, there are so many criteria are there. The inspection team will come and see your facilities, and then they will certify. And product licensing, you will have to apply to truck controller, and you will have to give all these paraphernalia to that. and with the label and the ratio of the material you are using, preservatives used, etc., etc. And all these Ayurvedic and herbal medicine, you cannot write that this heals a particular disease. See, you cannot claim that because there is no proof, actual proof is there. While allopathic medicine, there are proofs. But here you cannot claim that this is good for a particular disease. But you can write on the symptoms, say this is good for sore throat, this is good for running nose, etc. Indirect way you can always write. Now this is the label of two of my products, and it's a big headache. Then other issues are manpower, technical personnel, and most difficult is labor force. You should have good labor, and many a times, the labor working with you, they learn all the technique and go to another industry. Then sanitation workers, etc. And second is loan license. This is much easier. You can go to a pharmaceutical manufacturing unit, then talk to them, and then you can rent a particular space and machinery, and they may give you their expertise. So in the label of your medicine, you will write manufacturing address of that manufacturing unit. And you can write marketed and manufactured by name, by your name. So they will give technical facilities, their people will be there, and the responsibility of the quality of the work will be with the manufacturer. And then third party manufacturing is very easy, but it has got its own problems. See, you will have to enter into an agreement with another very reliable manufacturing company. So they will manufacture your medicine under their name. They will get license under their name. So their name will be written in the bottle as manufacturer, and you will sell it and marketed by so and so. Say my product manufactured by Paras Herbal Pharma in Vadodara and marketed by Dr. Daniel's laboratory. Now here the problem is that many people, they come to me that, sir, I want to manufacture a particular medicine. But I said, what are you doing? Sir, that is a secret. We cannot disclose. And no manufacturer, no good company will manufacture your product. If you say that, they will produce the medicine and give you. No, because the quality of the product is the responsibility of the manufacturing firm. So they will be in jail if there is any problem. Many people, they add allopathic medicines into it, and all those problems they are doing. So you will have to tell them that what exactly you want to do. So if you have a secret formula, it will never remain a secret once you tell them that this is what you have to do. Now, the problem you will be selecting, say, for diabetes, that is, India is a diabetic capital of the world. And suppose you want to prepare a medicine for diabetes, which plant you will be selecting? I have read a book that is published in 2000, and more than 1,800 plants have anti-diabetic properties. Now, of which 150 to 200 are very good. And out these 200 plants, which one are you going to select? And that is one thing. Second is adulteration of raw material. That is 80%, more than 80% raw material you are getting is adulterated. Then there is extraction procedure. You should know, I was sharing with you how to extract these things. Then lack of knowledge on active components, synergism, biocatalysts, antioxidants, etc. Because if you want to export this one, you will have to tell all these things to them. And they should be scientifically correct. Then only they will believe. So it is not that easy. Now, this is the problem of plenty. I told you about the anti-diabetic portion. Now, the plant you selecting should be with a large number of clinical researchers. Clinical researchers means, people have experimented on living patients with your medicine. The patients under strict watch, they will be given your medicine. Then a group of experts will be monitoring them every day, their progress and their healing which they are getting. Everything will be monitored, and the results are to be plotted and submitted. That is the clinical researchers. So if there are a lot of clinical researchers, select the plant which is having maximum amount of clinical researchers. And it will be better if you go for plants having more than one pharmacological activity. Then you will have to select plants which are commercially available, and they should be cheap, cost factor. If it is very costly, your medicine will be very costly and nobody will buy it. Now, I'm giving you the list of best plants based on my book only, but I do not have enough time to go through it. So this is something for all diseases. Now, we cannot reach international market because our products have poor selection of plants, adulterated raw material, inefficient extraction method, all those things which I have already said. Now, medicinal plant basic, your one extract will contain 400 to 500 compounds. That is metabolome. And many of the common compounds are pain relievers like gentisic acid, ferulic acid. Some are anti-inflammatory like ascorbic acid, cinnamic acid. So every plant is having some of these compounds. So every plant and every extract will have many of these compounds. Then coriander and licorice contain 20 antibacterial compound, and licorice contain 33% bactericidal compound. All these things you should know. And one drug plant will have many different actions. For example, Punarnava, Bravia. It is having punarnavoside, which is antifibrinolytic. Then hypoxanthine, it is hypotensive. Then diuretic compounds are there, antioxidants are there, ecdysterone, anabolic steroids are there. They are rich in potassium. Selection of plants are very important. Clinical researches are important. Now, this is about Vijayasar, Pterocarpus marsupium, and clinical researches are just shown there. Then the question is whether you go for monoherbal formulation or polyherbal formulation. Whether your medicine contains one plant or many different plants. What people are doing that they will just see the plants used by Dabur or Himalaya or anybody, or they may even copy my formulation also, change the proportion and produce a new formulation. Now, people say, you add this also, you add this also. There are so many people coming to me saying that suggestion anybody can give. Why don't you add this? Why don't you add this? But if there are 20 plants in a medicine, each plant will be represented by only 5%. So one plant 5%, second 5%. So the efficacy of the best herbs will be very less. So many times you can go for monoherbal. I have one product with only one plant, one product with two products with two plants. It depends. Then this adulteration, biggest problem. See, I am giving you some example. This is Brahmi, Centella, Manduk Parni, Bakopa Maniri. And this is very costly. Centella asiatica in Baroda, you get 1 kilo of dry Centella asiatica for 2,000 rupees. Now, all over India, this plant, Barimia marginata, is sold as Brahmi. See, this looks like, the second one, you just see the leaves of Marimia emarginata. And this is that of Centella asiatica. People do not know, the leaves look alike. And this Marimia is only 50 rupees kilo. So people say that why should you spend 2,000 rupees a kilo? While, and they are not bothered about the quality or the components. Then another biggest problem is Ashoka, Sita Ashoka, Saraka indica. And this is not available here now in market. I have stopped producing my excellent medicine because I do not get Saraca indica. Instead, you get polyalthia. The polyalthia is known as Ashopala. So people say that yes, this is also is Ashok, and Saraca is also Ashok. And some people, they use Bohemia variegata bark, Bombax Siba bark, and Kottakkal Ayurvedishala, they have come out saying that Shoria robusta is as good as Saraca indica. And this is Polyalthia longifolia. Now this is Shankapushpi. Controlled as pluricalis, evolved as alcinoids. They are all good. They both are good. But then Clitoria also is used as Shankapushpi. People supply it. Canscora also is used as Shankapushpi. Then another wool, actually it is semidesmus, but in market you get Decalepis, Echinocarpus, and Cryptolepis. And passion bed. Bergenia ligulata is the original passion bed. But then a number of other plants, Zeroa, Hamania, they're all used as passion bed. Then Gokhru. See, there is Tribulus terrestris. This is the small Gokhru, and Pedalium is the big Gokhru. I've just concluded, my one paper has come out. Pedalium and these two are entirely different. These two are entirely different chemically. Tribulus contains saponins, while Pedalium contains mucilages. And Tribulus contain flavonoids, Pedalium contain flavonoids, and none of the compounds are common. But still, people use Tribulus in place of Pedalium and Pedalium in place of Tribulus. But then there is another plant. This also having two horns. There is Acanthosperma hispidum that also is sold as Gokhru. Then the Shmula, the biggest problem. See, there are people do not know that there are 10 roots are there. Of which the first 5 are herbs. Now to get 20 kilos of roots of Desmodium gangeticum is almost impossible. So what they do is that they take the whole plant, they say Samula and Gokhru. They use Samula, Solanum indicum. They take the whole plant. Now we have just completed study of Gokhru roots and Solanum indicum roots. They are entirely different from the aerial parts. So then what are you going to manufacture if they do not contain the actual Dashmula? And the other 6 are trees. And to get roots of big trees is very difficult. Now, you should, many times, when you get the plant, you should be identifying by how will you identify? Now, see that no 2 plants are alike, just like 2 people. They are different. They are different in morphology, the anatomy, and in chemical constitution. You can look for the organoleptic characters. You can go for the micromorphological characters, glands, hairs, and you can now, this is trichomes of different types of bella. Bella, Adibala, Nagbala, they can be just identified based on the trichomes and different species of Indigofera by this midi-fixed hairs, and different glands are there, and other anatomical characters are also there. Then phytochemical analysis. Now, I have just before about 4 years, I have come out with a book on analytical methods of medicinal plants and economic botany with my son. He is a professor of chemistry. Now, this is published in 2016. It is sold out, and a new version is coming out by scientific publishers. And this is an excellent book on TLC analysis, plant drug analysis by Wagner, Blatt, and Skainsky, but it is very costly. And they get actual photographs in this. See, for every medicinal plant, mostly European plants, but Indian plants also say they are adding. And this is, you can compare the chromatograms. See, the left is the original Vijayasar, Pterocarpus marsupium. I had some doubt about one plant. So I did the chromatography with the original plant. So you can see this is in UV light, this particular fluorescence. So this is how we check. Now, cost factor Now, if you go to the market to buy this mal, this raw material, they will ask you, do you want super mal? That will be very costly, maybe kilo 400 rupees. And then you say it is very costly, then say you go for sada mal, ordinary mal. That may be about 250 rupees. And if you say, yeah, no, that also is very difficult, then they'll say that, sir, you go for chalu mal, kandam mal. And this will be very cheap, maybe 50 rupees per kilo. Most of the manufacturers, they will go for the normal chalu mal. And extraction methods, I already said, extracting solvents. You can use water to be extracted 3 times, or you can, water insoluble compounds are there, you can extract them with isopropyl alcohol. You just see what is the need for extracting with both water and isopropyl alcohol. This is Curculigia orchids. The left is the compound soluble in water. If you boil them in water, only these compounds will come. These many compounds will not come in your water. Now, this is Tribulus terrestris, Chlorophytum borevillianum, say Vidania somnifera. These things will come in water, this will not come in water. Now, then which one needs to be? So what I do is that for many of my compound, I extract them first with isopropyl alcohol and then with water. Then active principles, okay. Then antioxidants you can add. Then you can add bioenhancers. Then other recommendations are try to put 500 milligram extract in every capsule for 500 milligram capsule. So I'm doing 500 milligram pure extract in every capsule. So my manufacturer says that, sir, what are you doing? You will not get anything. What we do is that what normal people are doing here, they will put 50 milligram of extract and 450 milligram of some other material, maybe original plant material, or the material remained after extraction. And then I add bioenhancer, that is piperine. If you add piperine to any of your product, and then the efficiency will be doubled or tripled. You can add antioxidants, then you can add adaptogenic plants. Above all, this is honesty. People will check into what you are. See, many people, they ask me, say, how your product are. I said, you go to my website, see my biodata. I was a teacher. I was an honest teacher. And that is why I came out with a less, assets. I was living only by my salary. Otherwise, people make, I was the Dean, Faculty of Science. I was the head department of botany. There were many chances. But I never went for making money from other weeds. That is why my students came to me after my retirement. They said that, sir, we believe you. See, this is more important. We believe you. We want to do business with you. Then I said, if your purpose is helping people outside to the society, then you join with me. So they are the backbone of my work. And they have given the name of my factory as Dr. Daniel's Laboratory. Now we have about 15 products. I think I'm taking.
Can we conclude here?
Yeah. Okay. These are our products. So you can go to my website. I'll just show you my website. And yeah, you just see this one. My website is www.drdaniellabs.com. And you will come to know all my products and how we prepare. You can contact us in this email. And you can also refer to my Facebook account, Maman Daniel or Dr. Daniel's Laboratory. Or in acute cases, if you want to call me, you can use my name, my number. But don't call me for silly things. If you have a genuine difficulty, you can contact me. If you want to start a factory, if you want to have some formulation, you can ask me, I can help you. And I'm sorry, I had to just rush up
Thank you.
Thank you.
Thank you very much for your presentation, sir. Our next speaker is waiting with us. Over to Vidyavardhini madam and Dikshit sir. Can you share your screen actually? Dikshit sir.
Sure.
You start sharing your screen, sir.
I'm just sharing.
Over to Vidyavardhini madam. Please, Vidya madam, kindly start introduction.
Yeah, sure. It gives me great pleasure to introduce Dr. Ashwini Kumar Dikshit. I have known him for the past 18 years. Both of us were UPSC selected, that is Union Public Service Commission selected assistant professors posted in Puducherry. And right after that, I have joined Telangana University, and Sir has joined Guru Ganthidas Vishwavidyalaya in Koni. And Sir is an associate professor in the Department of Botany in Guru Ganthidas Vishwavidyalaya Koni, Bilaspur. And his qualification is MSc PhD from the University of Saugar. And he has 28 years of teaching experience and 25 years of research experience. And his area of specializations include medical botany, pharmacognosy, biotechnology, biodiversity, forest ecology, spiritual botany. And he has guided around 9 MPhil candidates and 10 PhD candidates. He has around 43 research publications, and he has published around 3 books. He has published around 5 book chapters, and he has submitted conference abstracts for 61 international and national conferences. And he has held various posts like Deputy Chief Protractor, Guru Ganthidas Vishwavidyalaya, Coordinator Anti-Ragging Committee GGV, Head Department of Botany GGV, Head In Charge Department of Forensic Sciences GGV, Member Academic Council GGV, Member Planning Committee GGV, Member Academic Council of other colleges, and he has attended around 47 seminars and organized around 3 seminars, and he has various interdisciplinary collaborations which include with JIPMER Pondicherry University, Satyabama University Chennai, Bharatidasan University Trichy, Pandit Ravishankar Shukla University Raipur, IITM Chennai, IGCAR Kalapakam, Tamil Nadu, Dr. H.F. Gaur University, Saugar, Madhya Pradesh, French Institute, the Pondicherry, Puducherry, and Bells University of Technology, Ogun, Nigeria. And I welcome you, Dikshit sir, and please start your session.
Thank you, Professor Vidyavardhini. Nice to see you after long. I think it's almost 10, 12 years I've seen you.
Yes, sir. Thank you. We know each other for the past 18 years.
Yes. So my topic today is actually, I have shared wrong. Anyway, I will start it. So today, actually, I'm going to tell about the immunomodulators from the herbal origin. And as we know that during this era of the corona, everybody is trying to boost the immunity, and they are searching for many things. As recently Professor Daniel told that there is a difference between the Ayurveda and the herbal medication and the herbal herbs. In Ayurveda, you cannot change the prescription, means what are the constraints are there, you have to go with that only. So development and evolution and modification of the drugs in Ayurveda is not possible. Whereas in herbal medications, botanicals or nutraceuticals, you have plenty. You can do anything in that. So I will start with this, that Brahma Arpanam, Brahma Ravira, Brahma Magno, Brahmana Hutam, Brahma Maiva Tena Gatyavyam, Brahma Karma Samadhina. So food itself is a Brahman, and Bhojan Mantra is a Brahman. The fire of hunger we feel is a Brahman, and we are Brahman, and process of eating and digesting the food is the action of the Brahman. It means we means of all the creatures on the earth, they are the Brahma, and they actually involve in everything. So we have to treat all the living creatures as a Brahma. Now, this is the mantra we are occasionally telling before as a Bhojan mantra. I'm not going to tell it. So health is health and wellness of the human being is actually dictated by the indication of nutritious food what we are having.
And these food, what we are taking in form of herbs or food, they help in combating the number of diseases we are getting from day to day. And as we know that at present, almost 80% diseases are the lifestyle diseases, because you are not taking the food in a proper manner, you are not using the herbs in a proper manner, and that's why you are compromising with your immune system. So this consumption of these biologically active ingredients through fruit, vegetables, and herbs, they are responsible factor which can cure cancer, cardiovascular diseases, even obesity, and many more. So this is another thing, that if you eat a sattvic food, means eating the agreeable food, and this will have the great impact on your lifestyle as well as on your health. So this is one citation from the great Valavur, mentioned the Thirukkural of the Tamil poet and philosopher. So actually, what we are eating, what food we are producing, and what in which environment we are living, so these, and public health, these are all deeply interrelated. So all these means if you have to be healthy, you have to look for all these four factors. Means your personal health, diet, from where you are getting the food, and the environment to which it belongs. So there is a very there is a very thin line between the diseases and the cure of the diseases. If your food is perfect, you can control many of the diseases or ailments without getting the real drugs. So this is the lifestyle I'm telling you. Bhojanante pivet takram, vasante pivet payam, nishante cha pivet vari tribhir rogo na jayate. It means you are drinking the buttermilk at the end of the meal, and milk at the end of the day, and water at the end of the night, means in the morning, you do not get the diseases. So this is the lifestyle. If you are adopted, you are able to prevent many diseases. Now come to the immunomodulators. So immunomodulators are biological or the synthetic substances that can stimulate, suppress, or modulate any aspect of the immune system, including both adaptive and the innate arms of the immune system. So immune system of a human being is having two wings, means it's adaptive and the innate type of things. So there are three types of immunomodulators, and they are immunoadjuvants, immunostimulants, and immunosuppressant. So immunoadjuvant means they enhance the efficacy of the vaccines or the drug, and therefore they are considered very specific immunostimulants, and they help us in getting the normal life along with the drugs. So means whenever you are on the drug, you have to take these immunoadjuvants, so they will stimulate the things, and they will enhance the efficacy of the drug. Stimulant means that they are inherently non-specific. They enhance the body resistance to any type of infections. So they just boost up your body. And third one is the immunosuppressant. See, we at present, due to environment and due to pesticides and many autoimmune diseases, and these autoimmune diseases are means here you have to suppress your immune system. If you will not suppress, that allergic reaction will come out, and this allergic reaction, due to the over response of your immune system, you are getting these diseases. So herbal drugs or herbs or botanicals can be used in all the three ways. Means they can help the boosting immune system, they can stimulate, they can suppress the immune system. So it was actually wrongly quoted by the many people that the Greek physician Hippocrates, he mentioned, let food be your medicine and food be your medicine be your food. But one scientist called Dinah Gardens in 2013, she said, she has gone through all the writings of the Hippocrates, but she didn't get it as coded anywhere. This is actually from the Indian origin, that let be food your medicine. And that's why we are using the Bhojan mantra before eating the food, or you are getting the, even for the preparation of food, there are mantras are there. So this term is going to the future of the drug industry, or to the health industry actually. The nutraceutical. Nutraceutical means when the nutrition and the pharmaceutical was much together. And Stephen D. Fallis, he has given this word for the first time. And this is used to describe a various type of product that can be used as a dietary supplement, and even they can be used to prevent many diseases. So immune responses requires actually many types of cells are behaving in a different time at different intervals within your body. So this microenvironments to maintain the immune homeostasis controlled by the different distribution and regulated expression of the cytokines. These cytokines are actually very important in giving the diseases. And you know that wherever there is a disease, there is some sort of inflammation inside the body. And this cytokines are the key target which can be treated well to treat the ailments. So that's already told. So these targeting cytokinins is now considered one of the logical approach for the prevention and treatment of the many diseases. Now, botanicals means these are the substances which we get from the herbs or from the plants original. They are the rejuvenators, they are the nutritional supplements. They have strong antioxidant activities, and they can help in overcome the oxidative stresses. And they operate through the immunostimulant, immunoadjuvant, and immunosuppressant activities by affecting the effector arm of the immune system, whether it's an innate or it's adaptive, both type of. Now, these synthetic immunomodulators are available in the market. They act, they work on the body, and they will help in getting the diseases cured. Side effects such as pulmonary toxicity, aplasia, increased risk of infection, hepatic fibrosis, lymphoma, nephrotoxicity, neurotoxicity, and so on, hypertension, renal dysfunction, kidney problems, tremor, hypertension, many more are there. And these botanicals are actually, they does not have any such side effect, because you are taking this as a food or as a food supplement. So these botanicals or the herbs are used to maintain the health and the prevent diseases. Now, these herbs or the botanicals, they play a very important role, a structural and functional role in mental and physical health of the human beings. And we know that these plants were used, or actually we does not have any specific system to reveal all this knowledge, but we are in a traditional system, whether it's a South India, North India, or whatever part of the India, or even I mean, in Asian countries itself. We are using the herbs and botanicals in the day-to-day life, and hence we are curing many diseases unknowingly just by adopting the tradition. So, see, these botanicals, they cannot be treated as a drug. There is means they can prevent from the diseases, but they cannot cure the disease. Once you have the disease, they does not have the potential to cure the disease, but definitely they have the potential to prevent the disease, and they also have the therapeutic uses in getting cured with the diseases along with the medications. So these botanicals can be obtained through various processes: extraction, distillation, purification, concentration, fermentation, and many more. So these botanicals may be of many types, means they have immunoactive botanicals. There are antioxidant botanicals which prevent cancer and also maintain the cardiovascular functions. botanicals which alleviate the psychological symptoms such as insomnia, anxiety, menopause, associated mood swings, and they also help in preventing many gastrointestinal problem and so on. So there are some chemicals which are responsible for cure, means which are having the potential to cure or prevent the diseases. They are simple phenols, phenolic acids, terpenoids, essential oils, alkaloids, tannins, flavones, coumarins, lecithin, and many more. So these are some standard herbal medications which are now used throughout the globe, and the list of phytochemical is also mentioned here. And most of these plants are having anti-tumor and immuno-related activities. And we know that there's the allium, aloe vera, Endographis paniculata, astragalus, curcumin is, everybody is talking about the curcumina longa. And they are giving, means sometime I have gone through some papers where they have mentioned that this curcumin is having the more efficacy than the control. How it come, I don't know. So mostly it is exaggerated one. So Nigella sativa is there, Panax ginseng is there. So ginseng, allium, and this Tinospora cardifolia, these plants are now getting wild popularity throughout the globe due to corona. So this immunomodulation activity is occurred through the phagocytosis stimulation, macrophages activation, immunostimulatory effect, and peritoneal macrophages, lymphoid cell stimulation. So these are some mechanisms, pathways through which this herbal immunomodulators works. Now, these botanicals provide the important nutrients that can help protect the disease against the diseases. They are rich in antioxidant, they are rich in omega-3 fatty acids, they are rich in fibers, and we know the user means applications of these antioxidants, omega-3 fatty acids and all that. And they help in promoting the growth and overall development of the body. Now, we'll have some specific type of foods which can be used as a immunomodulators. So there are potassium-rich foods are there. See, banana, potato, yam, spinach, and beans, lentils, kidney beans. So these are the plants which have rich potassium content, and this potassium is electrolyte which helps in conduct of the electrical charges in the body, also helps in the muscle contraction and maintain the BP and all that. Beta-cyanins, they are now used in anti-cancerous property they have. They are good for brain, they are neuroprotective, they are nootropic medicines, they are good oxidants. They protect the lipid profile by increasing the LDL and protect the membrane lipids. They protect liver from the toxic chemical pollutants, which we are prone to get every day. They help in digestion and also prevent the lung and skin cancers. Now, if we talk about the anthocyanins, we can see that they help increase the night vision. They help in reducing the cancer cell proliferation, and they inhibit the tumor formation. So actually, these anthocyanin, beta-cyanin, potassium-rich, these are all the common ingredients of the herbs, and they are present in almost all the herbs in different concentrations. So they prevent obesity and diabetes. They have the anti-inflammatory properties. And this anti-inflammatory properties is the most, they are making is the most important herbs, because all the diseases are associated directly or indirectly with the inflammation. Omega-3 fatty acids, we know that there's only fish is the source for the omega-3 fatty acids, but now there are plants which have omega-3 fatty acids, and these regulates the cardiovascular function. They have a role in many things, means anti-clotting blood and all that. Even the rheumatoid arthritis is a very common disease nowadays. And because we are not doing the physical work and physical exercises, so we are occasionally getting this rheumatoid arthritis. And omega-3 fatty acids helps in that prevention of the arthritis. Phenols and phenolic compounds, they are antioxidants, and these are the compounds which can help you in getting cure from the all viral diseases, as well as they will help in preventing the cancer, all type of cancers. So how they work is not important, and I know that we are having less time. So glycosides is another group of compounds which is having very important role in immunomodulation, and they are available in the Endographis paniculata, pigoriza, we call it, and most common is anthraquinone. Glucosides are available, and these glucosides are actually contributing to a drug value of the plants. Now I'll take some examples, used to improve overall health and promote the longevity. It relieves the stress, and as we know that the stress is the main cause of many diseases, and it is also having the antioxidant properties. And ashwagandha is having great immunity boosting qualities, and it increases the WBC count and natural killer cells. NKCs also, it will increase the number of these cells, and hence it is means ashwagandha is a very good herbs, and we have to use it. Bacopa, as I told that Bacopa now, Merimia is used in place of Bacopa in the name of Brahmi, as Daniel sir told. And this Brahmi, because see, nowadays we are using more brain than the body. So you need to improve your memory and factually recall the things at the right time. And this Bacopa Mondi is the answer for that. So this is the nootropic supplement. And in Chhattisgarh, from where I am speaking, is we are using this Bacopa Bhaji as a regular dietary supplement. Means here it is available in the common market. In the vegetable market it is available and people are using it. Broccoli is another botanical which is now used because it is having antioxidant, antimicrobial, and it's a good source of fibers, prevent cancer, improve immunity. So broccoli is another herbal botanical which is used. Chlorella as algae, we know that it detoxify the heavy metals. See, most of the problem with the present era is the pollution, the pesticide, the insecticide, and lifestyle. So these three things are the cause for all type of diseases. And if your liver is functioning well, means your liver is able to detoxify your body on the regular basis, you will not have many diseases. Similarly, if you are getting all the micronutrients and important phytochemicals which boost your immunity and make the balance between the tridosha, vata, pitta, and kapha, then definitely you will not have, means you are preventing the diseases. So these herbs are very important in doing all that. Green tea, common. Now this is the important thing, just I wanted to tell it. Nowadays the onion skill tea is available in the market. Especially in India it is not popular, but in some portions of the European countries they are started using it. And this onion scale tea means just the scales of the onion, the dry scales, you have to miss whatever we are doing. Whenever we are using the onion, we are just throwing the scales in the dustbin. So here I am suggesting you that you just keep this dry scales aside and let it dry further. And they are the best antioxidant known. Even the green tea means it is having many folds, much flavonoids than the green tea. And I have given the example here, means what is the concentration of the flavonoid is mentioned here. And this protect the liver, increase immunity, prevent infections, cure in stress and help in combating the cancers, decreasing the prostate enlargement, and also helping in getting rid of many cardiovascular problems. So this onion scale tea is another thing which can be used as a herbal immunomodulator. Moringa oleifera is a very important plant, and it is rich in all sorts of vitamins, magnesium, potassium, calcium, proteins, and iron. So this helps in maintaining the essential micronutrient requirement of the body. And this moringa is having 4 times the vitamin A than the carrots, 7 times the vitamin C of the orange, like that it is mentioned. So you can compare that freshly and dry leaves. Both are having, means you can use it as fresh or you can use it as a dry. Means what you have to do, you are just dry the leaves of the moringa, make a powder, and just 1 spoon of the dried powder of the moringa, add in the curries. Whatever you are cooking, some vegetables or something, you just put 1 tablespoon of dried leaves powder. In that, and you will you can see that it is having 10 times vitamin than the vitamin A than the carrots, 17 times calcium than the milk. So in this way, you are curing many diseases. You are preventing many diseases. So this is olivia. These are the various activities of the moringa oleifera. Noni juice is also now very common. Professor Madhivanan from the Chennai, he has promoted this plant for a long, since 25 years I am seeing that he is promoting this plant. And now this plant juice is available in every medical shop across India. So this is very important plant. And turmeric, as I told you, whatnot, everything, it cures everything. It helps in getting everything. And the beauty is if you go for the traditional type of things, you can see that, and grandma said that you just take 1 spoon of turmeric powder and you just roast it over the flame, and then take 1 spoon raw turmeric, mix it and eat it. Means turmeric is having a different potential. When you take this turmeric with the milk, it is having different function. If you take this turmeric with the fat, means ghee or neya, it is having a different function. And if you take this turmeric in the water, it is having different. If you take it as a turmeric, it is having different function. So the function depends on the mode of consumption, how you are taking it, and in which quantity you are taking it. Stevia, sir told that he tried to add the stevia and the chavanpras, but he didn't get the permission for that. Professor Daniel just now told. And stevia is a natural sweetener, and it can be grown everywhere very easily, and it is the best answer. Means most of the diseases we are getting is because of the white sugar which we are using daily. So if we replace this white sugar with the stevia, you can prevent many diseases. And this stevia is is growing like a weed. If you just plant it somewhere in some wide area, and another month you can see that plenty of stevia plants are available there. And you can take the leaf, you can dry it, and you can make use of it forever. So it is having benefits over many things. It also helps in managing the diabetes, which is another important disease at present. It helps in treating with eczema and dermatitis. By lowering the sugar, you can alleviate the preventive nature of the body. So these botanical, and they have associated with several powerful health benefits, and they not only prevent the nutritive deficiency, but they also protect you from various diseases, and they promote the overall growth and development of the body. So herbs are the answer for many things. In conclusion, I will say that the botanicals, nutraceuticals, and herbal medicine, they present in most of the funerary ingredients in a varying concentration, and the concentration, time, duration of the supply of the nutraceuticals influence the human health directly as well as indirectly. Manipulating the foods with the concentration of the active ingredients can be increased. Means, as I told you, just take a dry powder of moringa and add in a curry, and it will boost your health and overall development of the body. Diet rich in nutraceutical and botanicals, along with the regular exercises, stress reduction and maintenance of the healthy body weight will maximize the health and reduce the health risk. So overall, I just summarize that the answer for many diseases means 80% diseases which you are getting in form of the lifestyle, in form of the diet, means improper diet and all that, that you can cure just by adding some nutraceuticals, botanicals, or the herbs in your daily diet, and you will be healthy. So thank you very much. And at the last, I will say that you just plant some trees and protect our environment. Thank you very much.
Thank you very much for such enlightening lecture, sir. We will surely follow in our daily life whatever the plans you have told, and I also suggest participants also to please make a note, and you can be healthy without any medicine. With this, I come to the next speaker, Hema from Australia, paramedical staff. Hema, can you listen to me, Hema?
Yes, I can hear you, sir.
Hema, you can start your presentation.
Yep. Good afternoon, everybody. It's a pleasure and honor to be with you all today.
Mute yourself, share the screen. Means
Sorry?
you will come in a full this thing.
Yeah.
Down there is a green share screen arrow, up arrow is there.
Yep. Is that okay now?
Yeah.
Okay. So good afternoon, everybody. My name is Hema. I'm one of a medical professional here in Australia. We came to Australia about 11 years ago, and since then I have been working in a hospital. So it's really a great honor to be with you all and join this conversation of emerging trends in health sciences or life sciences. I have been listening to all the great lectures, and it is indeed a very informative session so far. I'm very glad that I got this opportunity to talk about.
Your sound is not this. You are not getting a sound, Hema.
Can you hear me now?
Yeah.
Okay. So as I was telling, it is a very informative session so far, and it was a pleasure to listen to some great lectures so far. So since we are discussing about emerging trends in life sciences, I think this discussion would be a little bit incomplete if we don't talk about the effects of the pandemic on our current life situations. So since you all know, like, we never thought that we would go through a pandemic situation in our lifetime, but here we are going through an actual pandemic, which is even as worse as a world war. So I'll just tell you a little bit about what information I have in my mind regarding COVID-19 and what's happening around the world. So COVID-19 is has been originally has come out from in December 2019 from Wuhan, in a province in China. So that's why it's called COVID-19. It's actually a group of viruses that's joined together, and which is immensely affecting the human respiratory system and the overall immunity of the body. So it has caused a very devastating effect on human society, global economy, and also global environment. So it is the most World Health Organization has declared this as a most urgent pandemic and a greatest challenge of the century and the humankind has ever faced since the World War II. So we can imagine the seriousness of this pandemic with all of us. I don't need to stress the importance of what it has done to all our lives, which is a sad reality. So the most cases, as we know, America is the worst corporate country which has faced the wrath of the pandemic with so many infections, and also the mortality rate is very high in America. Next comes the Europe and Southeast Asia. Although in Southeast Asia there are so many infections, the mortality rate is not as high as it is in Europe and America, probably owing to the immunity levels or the warmer weather, whatever it may be, but we should consider ourselves a little bit luckier than the rest of the world. So if we talk about the effects of this pandemic on all over the world, there are so many important things that we can count on our fingertips. The first one is the economical loss of the whole world in the form of shares. So all the companies are big shifts in stock market, where all the companies invest their profits has that has affected and the shares value has been lost. Not only the big companies, even the common man has lost a lot of their money in the form of shares, because if people go and work in a company, the company pays superannuation. So all the superannuation is held with a third company who invest in shares. So even here in Australia, like I myself, I might have lost $20,000 to $30,000 in the whole pandemic. So that's how everyone has lost the money. That's the biggest long-term problem we will see for some time. And also the second effect that has caused is a difficult year for the job seekers. Like if you are a prospective graduate, came out of the uni and wanted to join in a job or anything, but the job employment rate is ever so low than what it was last year or the year before. So millions of workers have also been put on government supported pensions or all those things, especially in the developed world, but probably not so much in India. There's not much great economic support from state-wise, but there's still very less job opportunities all over the world. And the world has all moved into a new recession era where each country is looking at how much damage the pandemic has caused and what ways it can do to improve their financial situation and improve the lives of the common people. That's the second one. The third important reason that I want to take up is the travel industry that has affected a lot. I can't travel to India if I want to now, so that's how it is. The travel is completely impaired, and it's very badly affected, with most of the airlines going into liquidation or selling the company to a different owners, or the new variants of virus that is emerging, the UK strain or whatever strain that is coming out, it's still a huge challenge, and they cannot predict a time when the normal international travel would resume, which is very unhelpful, leave alone the monetary loss of the people. But if we don't know when you can go and see your loved ones, that's a very tragic and terrible situation that we all are in, which is very unfortunate. The next area that has affected most is the hospitality area, where all the supermarket, you can't probably go and shop in the supermarkets as earlier. So we have to, most of them went on for online shopping, and the hospital industry like restaurants and food serving businesses, they have lost their business, and most of the people are shopping from home at the moment, which is kind of a new trend, and some people like it, some people don't like it, but there is a change in the shopping behavior recently. So yeah, it is a new trend that we have to accept. Next is the pharmaceutical companies are one area that they have benefited the most from this pandemic, I should say. So governments around the world have pledged billions of dollars for COVID-19 vaccine and treatment options. So everything, these researches have been going on for the past 1 year. So all the governments have paid for these companies to manufacture a drug more suitable to that area and the temperature. So yeah, there we are. So that's why we have 3 or 4 different kinds of vaccines in all over the world going on. There's Novavax, AstraZeneca, and also there's a Pfizer vaccine going on around. Now they don't have 100% efficacy, or they can't prevent the disease 100%, but they can provide some level of immunity. It's like an influenza vaccine. If you take like in Australia, we take influenza vaccine every year, but it doesn't provide 100% protection to influenza virus, because each year there may be 2 or 3 different strains of influenza virus that might affect. So even if you're offered protection in strain, so that is one thing. So yeah, so since this is all a very big change or effect of pandemic on our lives, we can also see about the per capita income of the families have decreased. So someone who used to earn well enough for the family, now they don't have as much as they need to, or I should say the savings of the families have come down, which is very unfortunate. And the other thing is we have to take into account the mental health of the population all over the world, which is like really gone to the rock bottom, and not many support services. Although even if the support services are available, people don't have the access to go and reach out for these services. So most of the people have ended up having domestic conflicts, domestic abuse, and also going into depression and anxiety. We don't realize how big a problem this is until we actually see or hear from someone that someone has hanged themselves because they're depressed. It is a true problem. It is a real problem that most of us underestimate. So mental health of the population of all around the world has taken a very steep decline downwards, which is very unfortunate. The next thing is, if with all the pandemic effects that has caused all over the world, all the countries are calculating of how much recession they are into, or how deep into the economic loss they are, and they are advising steps and processes to overcome these. Like here in Australia, the government has pledged a multi-million-dollar assistance, step-by-step assistance to most of the industries and businesses, and also individuals who have lost their work and who are not keeping healthy because of the COVID. And yeah, so for that reason, I hope the same in India too, that the local government or the state government is reaching out to the people to help in as best ways as they can. So in this situation for us, it's important to be together with all of us as a family, and reach out for the friends and look after each other. It's a very tragic situation that we are in. Hopefully the effects of pandemics are not as worse in India, because India is a self-sufficient country. if you take other countries, if you take an example of Australia, it's not a manufacturing country. It can't produce all the things that we are using. So we have to import all the things from China or Bangladesh or India or all the other countries, because the labor is very expensive here. But it's unlike this situation. India is a self-sufficient country. India has its own food supplies, minerals, production industries. So I should say that the global economic effects of COVID would be a little bit not so worse on India compared to the other countries. At least it's the responsibility of the government now to take out, to go out of their way and help common people, and to make sure that they are receiving adequate support in health, education, mental health, and so such a sad thing that all the education industry is how bad it has been affected, with the kids not receiving the academic information or education that they needed for the last year, and also teachers losing their jobs and losing their motivation. So it's all of our responsibility to support each other and keep us all in loop. Even if there is any support that is being available or offered, we all have to help each other and reach out for the needy. So I have seen how the pandemic is managed here in this country, like going on for very strict rules of lockdown, like we were not even allowed to go out of the house except for a couple of reasons, or even if we are allowed to go out of the house, we are only allowed to go into a 5-kilometer radius. You can either go for shopping, the essentials, or the petrol, or a medical facility, and that's all. So we were here like in lockdown for quite a while. For at least a couple of months each time. There were 2 waves here, two surges of COVID cases here, and both of these times we went into lockdown for a good couple of months, which have controlled the disease. The thing is, this kind of lockdown doesn't work in India, or has less effect in India, because of the nature of the population and the lack of facilities in a 5-kilometer radius. If you say someone in a village to not go out of 5 kilometers, he wouldn't even cross his village. So that compared to the vastness of the area and also the facilities, it may not be possible in India. But what I'm hearing from all there is people are trying as best as they can to maintain social distancing, wear a mask, and and we can use the benefits of the medicinal plants and foods that all the knowledgeable people have described now, and use them to boost your immunity and keep up your better health and look after your health. And it's as important to keep ourselves in loop with the emerging trends in life sciences, as it's a need of the hour for us to be in the loop and keep ourselves informed. So I really thank Dr. Haleem Khan sir for giving me this opportunity to be on platform with you all guys, and it was such an honor. I was his student. I was a student of microbiology a long time ago now, but it is an honor and a pleasure to be with you all. I wish to join and hear further seminars and conventions like this, and I would make it as a message to reach out to all the students now to keep up, do your best, achieve whatever you want to in your life. You will do it. You'll definitely do it. You just need that effort, that you believe in yourself, and you just work towards it, and you will be there. Thank you so much, sir, for giving me this opportunity.
Dr. Nandikar, can you start sharing your screen, Dr. Nandikar? Yeah, over to Dr. Jalandhar. Dr. Jalandhar, can you introduce our next speaker, Dr. Mayur Nandikar?
Okay, sir. Thank you, sir.
Dr.
Very good afternoon to all. Sir, very good afternoon to all. I am audible, sir?
Yes, you are audible.
Hello?
Yes, you are audible.
Okay, sir. Thank you, sir. Very good afternoon to all. It's my immense pleasure to introduce Dr. Mayur D. Nandikar, sir. He has completed his MSc Botany from Mumbai University, Mumbai, and MPhil in 2009, PhD in 2013 from Shivaji University, Kolhapur. He is specialized in flowering plant taxonomy, nomenclature, biodiversity, and conservation. Now, he is presently working as a scientist and head at Nauroji Godrej Center for Plant Research, Shirwal, district Satara, Maharashtra. He has been worked as a postdoctoral fellow in the year 2013 to 15 at Goa University. Then he got a Young Scientist Award in 2016 to 2019 from DST, India. And also he has total 6 years of teaching and research experience in plant taxonomy and conservation. Then see, he has worked as a project fellow funded by Rajiv Gandhi Science and Technology Communication from July 2009 to August 2012. He has completed 3 projects and ongoing are 3, and these projects were funded by DPT Science and Engineering Board, New Delhi, Godrej Foundation. He has described a total 5 new plant species, and he is working as a reviewer for many scientific journals from different countries. Then he has presented many papers in national and international conferences. Also, he has published a total 33 research articles in national and international journals. He has wrote 2 books. First one is a taxonomic revision of Indian Commelinaceae and Field Guide. Second one is Commelina for Flora of West Bengal. And see, he is a life member of Bombay National History Society, Mumbai, Indian Association for Angiosperm Taxonomy, Calicut. Then he has visited many countries such as UK, England, Royal Botanical Garden, Kew, and National History Museum, London. Sorry, Natural History Museum, London. Then Dubai, Saudi Arabia, just like these countries he has visited. Thank you very much, sir, for giving me this opportunity. And I welcome Dr. Nandikar, sir, to deliver your talk, sir. Please.
Yeah, thank you, Jalendra Saab. Thank you, Dr. Khan, for providing this opportunity. It's always good to have the talk on family, which I most like, which is very close to my heart, family community. I just joined before half an hour on this group, and I found the emerging trends in life sciences. I don't know how it will be going to help, because I really don't know the audience what exactly they are expecting from my side. But I feel we should not exclude the taxonomy, because it's like subject that everyone likes. I feel whatever we are doing these days, I mean to say from the from the birth till death, whatever we are abstracting, all are things from the plants. And we should not forget the contribution that given by the different people in the field of taxonomy. And this is what I wanted to emphasize over here. Although many of the emerging trends are coming forward, many of the evidences that found that applied aspects are more, I mean to say, having the bloom compared to the basic sciences like a taxonomy. But still, I feel we are like a plinth on which everyone can build the building. So with these words, I will start with the Commelinaceae. I don't know how many of you are aware with the family Commelinaceae, but it is one of the diverse family, and it is one of the commonest family in India, and as many of the members that occurs cosmopolitan, or some of the members are very restricted to the particular habitat. So what I am going to today deliver that how the commonality, my interest has been flourished, how the taxonomy varies in the within the family, how the characters, characters varies within the family, and secondly, the importance of the plant species according to their endemism, as many of the species are endemic to the Western Ghats as well as some are endemic to the northern eastern parts. So I will talk on the taxonomic aspect as well as I will share some of the slides on the endemism. Since 15 years, last 15 years, I'm spending much time on the commonality taxonomy. And as time moved, the classification has been changed. As earlier, we used to call it as a dicot and the monocot, but now we are talking about different clades. So commonality has different families again, and into that the commonalities, that was the order it has been placed. And now the many trains has been evolved. It's only we are using, but in addition to the taxonomy, we are important aspects that we are using the palynology, we are using anatomy, then the biochemical phytochemistry, and epidermal characters to find out where the family has been placed. So these are the different classification. This is, I have put it from the very modern classification of Angiosperm phylogeny group 4th. As I was telling that commonality has like a commonalineals has different families, and one of the diversity you will find in the commonality, as we popularly called as a spider-bird or the day flower, as the flower lives for a very short period. Therefore, probably we used to call it as a day flower. Or often, some of the genera we used to call a dew flower. As I told that they bloom for very early morning hours and they fade by the noon. So total in the world, 41 genera are there and the 650 species. See the diversity within a small family. As many of us are aware with the Tradescantia, some of may aware with the Commelia bengalensis, cyanotes and all. But if you see the diversity, we have many garden ornamentals, household plants, and many of them are endemic now. And I will come to the diversity in the morphology. But this is the overall distribution of the commelinacea. If you see, Commelini, the tribe Commelini is dominant in the old world, whereas the Tradescantia is dominated in the new world. So this says that how the species has been diversified and speciation has been happened. If you are asking me about India, we have like a major diversity of the Commelini in Western Ghats, whereas Tradescantia is more dominated in the northern eastern parts of India. And if you see the number of species, total number of species occurs in India, almost 100 species we are getting in India. Out of that, 26 species are endemic, and some of the intraspecific taxite also has. So this is overall classification. So if being a layman, no, you can calculate the, analyze the family while seeing the flowers. If you see this flower, you can make out 6 prominent dots into it. These are the 6 fertile stamens, where in this, you may not get that 6 typical dots over here. It means that it may have some additional character called staminodes. So Tradescantia characteristically segregated due to having more symmetrical petals and 6 fertile stamens, whereas the Commelini is having 3 fertile as well as the 3 staminodes. So these are the genera listed listed under the Commelini. The Tradescantia occurs in India. And see the diversity here. If you see, many of the species are having the different habitat. In fact, some are like showing a climbing habitat, some are growing erect, some are like a procumbent, some are having the ascending, some are like again the procumbent ascending, and some grows prostrate. So if you see the diversity in the habits, you will find many habit diversity into it. In fact, if I'm talking about the roots also, if you see, some are having the fibrous root, very thin fibrous root, which is characteristically occurs in monocots, but some are having the thickened fibrous root. I'm not talking about the tuberous, but here you will find the fusiform tuberous root here also. So it diverges in habit as well as in the root characters. Similarly, it also diverges in the inflorescence. If you see the diversity in the inflorescence, it's amazing. You will get the axillary flowers, you will get the terminal cincinae, you will get the fasciculate, reduced fascicles. In fact, you will find the axillary or the leaf opposite fascicles too. And most common type is like having the thyrosis or the cincinae of multiple flowers. It may arranged in the lax panicles, or it may be in the reduced cymes like this. And Belosynopsis is one more genera in the family, is the genus in the family Commelinaceae, which has umbellate cymes also. And if you see the Commelina, which is very characteristically, the flowers or the inflorescence are enclosed in the spathe. See the diversity again in the flowers. So flower color is there, shape of the petal is there, the staminodes, arrangement of the staminodes, then the style, or if you see some of the flowers are having a petaloid sepals or the sepaloid petals you can call. So this is what the diversity you can find within the family. You see the striations on the petals. So these are very unique to the family. See the capsule. Again, the fruits are having a very diverse situation. If you see, diversity is there. The typical trilocular capsule is there with the 6 number of seeds into it. Similarly, bilocular capsule with some kind of farinose attached to it, which shows the appendages. Similarly, some of the berries, it cannot call as a berries. It's like in between the berries and the capsules with like a typical metallic blue color or the black color, you can say. And this is bilocular again with the humpbacked back. So this is the diversity you will get it in the Commelinaceae. If you can see the seeds under the scanning electron microscope, you may find these beautiful structures. For example, if you see the taster of these Commelinaceae, it has a reticulation. In fact, this position of the embryotega also matters to classify the groups. For example, terminal embryotega, you will get it in the genus called Tradescantia, Cyanotes, Tradescantia, Belosynopsis. Over here, in the Rhophalophora, it may the semilateral, similarly with the semilateral in the Mudania. And the position of the hilum also varies. If you see the hilum positioned over here in the ventral part of the seeds, and see the shape of hilum. If it is the linear one, or it is, you cannot gauge the shape. It may be a micronate probably, or it may be a elliptic over here. And some are having the typical pits on their ventral side. So seeds are also important, and it can be used as one of the reliable characters. I will just go quickly because half an hour is not enough to describe entire group or the entire family, because it is very diverse, as I said. So quickly, I'll go through the alphabetically what the genera that we have organized. So in that way, you will have the glimpses of the different genera occurs in the falls under the family Commelinaceae. You may aware with the, as I said, the Commelina, Cyanotes, Tradescantia, but some of the genera are very exclusive, and you may not aware as it grows in very, restricted areas of the core part of the Western Ghats as well as the northeastern parts of India. So I'll just move quickly through the genera. This is the Amiscotoloid, which has characteristically axillary flowers, and these are in the fascicles, and the sepals are succulent. Total 3 species we are having. Entire world 26 species are there, and one of the species is endemic to India. So this is the Monosperma, which grows in Nicobar Islands and which completely grows like a postage, and having the decumbent kind of habit laterwards, which produces the terminal leaves. Another important genus is Belosynopsis, which is very commonly known because of it's having like a epiphytic on the lithophytes also. So one species is endemic, vivipara, because it grows on the tree trunks and it produces like a vivipara in what we are getting in the mangroves. The mother plants will grows on the plant, seeds will grows on the mother plants, and from the next the new plants will come up. So this is the typical species grows in the Western Ghats, and the other species QNC is also cultivated for its ornamental importance. This is the QNC, which has beautiful vinaceous leaves, and it is very popular among the European gardens because it has the ornamental leaves. So this is the illustration of the Belosynopsis cuensis. You may find these plants in the nurseries as an ornamental. Two species, it's popularly basket plant as people used it as a basket plant. And this looks like a bromeliad. This is the Calicia. People also popularly call it as a Spironema. And one of my favorite group is Commelina, where you will find a big diversity into it. Total 170 species you will find across the globe, and 27 species occurs in India. Out of that, almost 7 species are endemic to this part of India. This is what the habit where it diverges. As I was telling, some plant grows erect, some are forming the mat, some are like growing the prostrate. See the diversity in the clistogamy. You may aware that clistoglamous flowers in Fabaceae, especially Arachis hypogaea, but here is another example that is from the Commelinaceae, where it produces aerial as well as the clistogamous flowers on the lateral shoots, which grows underground and produces the clistogamy. Similarly, leaves and seeds. As I said, the sheaths are very peculiar. For example, some sheaths are open, some sheaths are closed, adherent to the stem. But see the leaves, some you will get it the linear leaves, you will get the ovate, oblique leaves, somewhere you will get the ensiform leaves. So tremendous diversity is there. Again, I'm come to the spathe, that is the unique character of the genus Commelina, where you will find the fused spathe, open spathe, lanceolate spathe, again the aggregate spathe. Here you will find the solitary spathe with the typical peduncle. Here you will not find the peduncle, the spathes are aggregate. So these are the characters that you can use to identify the Commelina in the field. Similarly, I have tried to open the spathe and try to show the inflorescence or the fruitation. So see the shape of the spathes, it's very diverse. And secondly, how it accommodate the capsule into it. So spathes are protecting the young flower birds as well as protecting the young capsules. So it works in a two different way. It's like a very advanced character that again you will find in the family Commelinaceae, where the protection and the security is more preferable. It cannot be, I don't have much time to discuss more on this, but if you see this image, you will find some of the organ is missing into the flowers. For example, I used to call it as a puzzle. This is the flower coming from the lower cincin. This is the flower coming from the upper cincin. If you will find the pistil is missing over there in the upper cincinous flower. It means this is the male flower and this is the female flower. This is what gives an aid for the pollinators to attract to them towards. So this is how the family has some advanced character. They are like a producing the mimicking flower, which appears to be a bisexual, but it is not. And actual flower, that the fertile flower, you will get it usually in the lower cincin. So these are the important characters. Once you are into the family, once you are into the group, you need to identify and you need to understand such characters to identify the plant families. Similarly, the colors, as I was telling in initial slides, the colors are actually varies. I have not shown the white color over here, but some of the plants produces white colored flowers. Similarly, it has ochre yellow, blue, then the purple, lilac shades. And similarly, the photographing Commelinaceae is one of the tricky job, because one thing, you have to be in the field very early in the morning, as flowers fade by the noon. So if you are reaching to the field by the noon, you will not get the flowers in the bloom. So that is one part you need to understand. Secondly, you need to understand the characters, that what are the characters which can be highlighted, what are the importance, having the taxonomic importance. For example, I have cut the cap space and I have shown the capsule inside it. Similarly, if you see the habits, so we need to very particular when we are capturing the habits. So entire things should be come into the one photograph. So these are the basic techniques that we need to understand while photographing the common linaceae. And see the capsule number, flower number. As I was telling, some flowers are exceptions. They produces the male fertile flower in the upper cincin. As the lower cincinus flowers and the fruit numbers, if you see, it has more than 9 capsules. And similarly, it has different flowers. For example, this is the example where it produces the fertile flowers. Here, also the fertile flowers. But in earlier photographs, there was a male flower and down there was a fertile flower. So this diversity is there. I will move towards another important genus called Cyanotes, which almost half dozen of the species grows in India, and 4 are endemic. It is very peculiar. Everyone can identify in the field if you know the circumscription of the genus Cyanotes, how it defined from the, or how it is separated from the Commeline. So Cyanotes has scorpioid signs. These are the scorpioid signs. And Embryotega, you will find it terminally. So of the plant, I'm holding the entire plant in my hand. So how small it is, and see how big it is. It cannot accommodate in my single frame, camera frame. And some of the groups are like very unique. For example, I was telling that Cyanotes is very unique because it's having the scorpioid sign. But if you see this plant, this is Cyanotes axillaris, where you will not find the scorpioid sign or that unique bracts on the like a falcate bracts and all. This is Cyanotes axillaris produces the axillary flowers. It may be in 1 or 2 or 3 numbers, but it doesn't produces the scorpioid sign. See, again, the puzzle will be always there. If you are comparing the 2 species together within the group, there will be many things that you need to understand. For example, in this, you may find the anthers. You cannot make out the anthers in this plant, the left-hand side plant, because it is black in color, where in this it's completely yellow or the lemon color. So these are small differences that you need to understand. For example, here the hairs, the moniliform hairs are colors, white as well as the lilac or the faint blue. Over here, you will not find the bicolored moniliform hairs. So what it says, in fact, see the peduncle, it's like a filiform, or you can call it as a very weak. Over here, it is complete erect and straight. So this is what we need to understand, the characters within the group and within the family. Dictyospermum is another important group which has 3 different species in India. One is known from the Andaman Islands and Myanmar. Other 2 grows in the Western Ghats, and both are like endemic to the India and Borneo. That means that both the species are 3 species. All the 3 species are endemic to the Southeast Asia. In these, if you see, there are the concept called thumb-eyed and the pin-eyed flowers. In this case, the flowers organs, for example, the stamens are like grows more and it covers the pistil. For example, if you see here, the pistil is here and it is covered by the 3 fertile stamens. Similarly, over here, again here. So it called the thumb-eyed flowers. And one more part you can observe over here, the petals are not much having the differs in the color than the sepals. It almost sepals are the petaloid, or petals or the sepaloid. If you can see properly, the sepals having some kind of highlighting, more highlighting than the petals. But in many cases, you will not find these differences. So this is the uniqueness that Dictyospermum has. And one more group that occurs in Commelinacea, that's Phloscopa. If you see the Phloscopa, very interesting characters is glandular sepals. Here, also here. Almost 20 species are there across globe, majority of the species grows in Africa, but Phloscopa scandens is one species that occurs throughout India, and it grows along the perennial water streams. Mudania, which is one of the diverse genus in family Commelinaceae, almost 50% of the species occurs in India, and many of them are endemic. So see the diversity in the genus called Mudania, diversity in the flowers, diversity in the inflorescence, diversity in the seeds, where you can find the pallium position as well as the embryotega position. Similarly, another genus called Pulia, which also has like a 3 to 6 species, almost 6 species or 5 species in India, total 17 species in the globe. And this group is characteristically identified with the help of its berries. For example, if you see the capsules, these are the very unique in their character. You will find the petaloid sepals again in this group, but the capsule is one of the important characters, and the seeds always biserate into it. And as I said in my initial slide, the capsule always produces metallic blue color or the black color. This is the Porandra, which occurs in Arunachal Pradesh, and recently it was described by Distillery from the Netherlands. And we were thinking that we are having only one species of Rhopalophora, but we are again adding one more species into it. It is having the characteristic of the glandular hairs on the capsule, humpback capsules, and seeds are also having the farinoles. One of the climbing species, Triptylirion, which grows in the northeastern parts of India as well as Bangladesh, Bhutan, and Nepal, and towards the China. And it is characteristically known by having the climbing habitat as well as the Commelina like a spathe. This is the Tinantia introduced long back into India, and now it is naturalized. You will find this species in Punjab, Uttarakhand, and other parts of the eastern India. You may aware with Tradescantia, which is very popular group for the cytology. People may use this plant as a garden ornament, many people are using it as the hanging basket plant and all. But what I am finding very interesting, this is the closest relative of the group called Cyanotis. If you ask me where the Tradescantia came from, it came from the New World. Particularly, many of the species are from the Mexico and Brazil. But now it's such a popularized throughout the world that everyone knows the Tradescantia. And Tricarpellima. If you see the Rhopalophora, which I have shown you earlier, that also has the similar kind of habit, similar kind of inflorescence. But here, the Tricarpellima different from the Rhopalophora by having many seeded capsules, and inflorescence usually lacks in thyrosis. So there is no much difference between the Rhopalophora, Anilima, and the Tricarpellima, but there are some minor differences that has been analyzed by the previous worker, and they have kept it this group on the separate label. So if you ask me what I did in last 18 years in the group Commelinaceae, we found some new species, we described some new species. This is what we have added from the Kolhapur, Maharashtra. This is Saddlepicensis, exclusively known from the Saddle Peak National Park, Andaman Islands. This we have added from Karnataka. This we again added from the Vidarbha, Central India. This is what we have added again from Karnataka. And this is again we have added Commelina littoralis, which grows on the littoral habitat of Andaman Islands, which is closely related to the Commelina caroliniana. See how beautiful flowers. This is what the interesting story it has, how it found in India. It was earlier known from the Sri Lanka. We have rediscovered this plant from the Chennai. And this is we have added from Andaman Island. It was like a discovery after 150 years. This is African species earlier known from Africa. Now we have added from India. So this is the journey that I wanted to tell you something about the Commelinaceae. I hope that you understood some of the species of Commelinaceae. Of course, it's a big topic, and it may require more than 1 or 2 days to understand the thorough taxonomy and thorough endemism in the Commelinaceae. But I feel if you have someone have interest in the studying the taxonomy, commelinacea is one of the group that where you can work for your lifetime, as it is more neglected because of the flowers are very directions. You cannot preserve the plant flowers through the barriers and all. You need to have the wet preservation. Still, it has a scope because it is one of the having the evolutionary significant, as many of the commelinacea is properly mainly used for their cytology. And if you see the karyotype variation, it is almost evolutionary. It's asymmetrical. That is evolutionary significant, and it is continuously evolving family. If you see the diversity in every form, if you take the habit, if you take the habitat, if you take the flowers, if you take the capsule seeds, it is very diversified. So what I will suggest you to read more about the commelinacea, try to understand the group, and if you really wanted to do something on the pollination biology interaction, or to understand the cytology, to have the molecular phylogeny and all, this is the very good group where you can work. So with this word, I will stop here. I thank you again that Dr. Khan has given me the opportunity to express my thought about the commelinacea, and thank you for the organizing such beautiful seminar. Thank you. Thanks a lot again.
Very thank you, Dr. Mayur. It was a long dream come true since 2019. It was keep on. We were tracking you, and at this moment we could see you on the online session. It was very interesting to see the beauty what you have. Actually, it is in your camera. The commelinacea has become so beautiful. I think it's a thing where how you describe this plant and how you describe the beauty of the flower and everything makes very special, really. How many times we see your presentation, the same presentation, even though say with some additions, it looks like it brings a great aura. And a person who don't love even flowers, who don't even like the plants, they will start liking by or they will turn towards your work, whatever work is there. You attract, you charm the people by making this looking so beautiful. So overall, it is a heartfelt thanks on behalf of the Department of Botany, Telangana University, to listen to you and to witness to you. And now you are in our video forever in an international conference after a long time of gap. You have accepted the thing. Okay. So thank you very much. And I request now Aruna ma'am to overall summarize today's event. Over to Aruna ma'am. Aruna ma'am, are you there? Yeah, you can summarize today's session and then we'll conclude.
Yes, sir. All the technical sessions have been closed, sir.
Dr. Ram Prasad is not doing fine. Dr. Ram Prasad is not doing fine.
Yeah, I thought maybe we'll be having his also. Okay, sir. Okay, sir. So as on today, as it's the first day of holding international conference on emerging trends in life sciences, we have come to an end after holding for 4 sessions, where we had 2 lectures in the morning session, technical session 1, and we are grateful to have Dr. Ramakrishnan, and we are thankful to him where he has shared his experiences all about plant bamboo. And the same manner, we are thankful to Dr. V.V. Belawadi from Bangalore, that is University of Agricultural Sciences. And his lecture was really interesting and it was impressive. And the title itself was a little bit difficult and different for us to understand. But still, we have learned how and why these insects play an important role in the mechanism of pollination. And pollinator-mediated speciation in plants was really impressive. And he has also told about the number of flowering plant species in India. And he has also told about flowering plants in the world, and out of which the plants in India were really, sharing the platform. And he has also told about the name of the book, and it was The Forgotten Pollinators. And one has to read this book to understand more about the mechanism of pollination. And sir has also told for efficiency of pollination is more by bees, and more than 2,500 species were in the world and 750 species in India. And he has taken one example from Gingiberaceae, and it was Litoria cardamom. And it was interesting to note that cardamom is a pseudostem. And he has also made us understand for floral biology in cardamom. And he has also told how the number of capsules would be increased, how the flower set could be increased. And this is really included in syllabus of our fourth semester students. We have one content fruit set. So we are grateful to you, sir. And next, following, we had technical session 2, where we had lectures by Indira Sakuran madam and also Professor Daniel sir. And we are grateful to both of them. And Indira madam, as she was a teacher and as she has worked at India and New Zealand, I think, she has really made us understand the role of a teacher in life of students. And it's really student-teacher relation. And it is a teacher who will build the life and career of a student. And it's happiness to a teacher when a student will grow to heights. And this is how she has made us understand the role of science in daily life. We are thankful to you, madam. And next, it was Daniel sir, and the topic was a little bit different. It was about business opportunities in the field of medicinal plants. And he was from University of Baroda. And all these years, we are teaching only about taxonomy and what about the families and how about the cultivation of the plants. But really has made us impressive. And this is the way where we can give life to students after completion of their PG or PhD exclusively in the field of science. So students think that there is no other way other than teaching or doing any other job. But it is as shown, you can also do business if you have got some knowledge of medicinal plants. And he also told that it's botanists who can supply raw material and who can also manufacture medicine and who could also manufacture the extracts from different plants. So this was really interesting. And we have also learned how Ayurvedic medicine would be different from herbal medicine. So we are grateful to you, sir. Thank you. And of course, in near future, we will be sending few of our students who have interest in this area. So kindly help and guide them, sir. This is our personal request from department side. And following, we had technical session 3, and it was Dr. Ashwin Kumar Dikshit. And he has told all about immunomodulators, and he has covered about immunoadjuvants, immunostimulants, and immunosuppressants. And from among the plants, he has very clearly told that ashwagandha is a plant which will boost the immunity and has got many qualities of boosting immunities. And during this context of pandemics period, of course, all the participants might have or would have noted the importance and significance of this plant along with all other plants which he has covered. So we are thankful and grateful to you, sir. And next we had a talk by Hemalata. She was from Australia, and as she's working from hospital, she has shared her experiences from her side, not only based on context of Indian levels, but also her experiences as living in Australia. So we are thankful to you, madam. And it was technical session 4, where we had talk by Dr. Mayuri Nandikar. And this was really, a stretched talk on family communicacy. And right from our intermediate standards, we'll be learning this Commelina bengalensis, where we have taught students, and where we have also learned that we'll have cleistogamy noticed in members of Commelinaceae, that is Commelina bengalensis. And he has very clearly focused the slides showing for diversity of different species belonging to family Commelinaceae. And as such, he has also told how many species have become endemic in India. So this is the time for us to conserve almost all the species belonging to family Commelinaceae. And he has also started his lecture with AGP system of classification. And this is to be noted by students and scholars who have attended the conference. And with this few remarks, I would like to close the session. I would like to close from my side. And I thank Haleem Khan sir for giving me this opportunity. And I request all the participants to show same interest and continue the tempo to attend for tomorrow's conference and be on time. Thank you. And I thank all the dignitaries and all the resource persons who have kindly accepted our invitation and who have shared the dais of Telangana University. And in near future also, I expect the same support from you. And we will be inviting you as many times. And let us maintain for cordial relations. And I thank Registrar of Telangana University, Professor Naseem Madam, who's continuously supporting the Department of Botany, Telangana University. In the same manner, I'm thankful to colleagues of the department. And it shows our unity in handling the programs. And as such, it is Dr. Haleem Khan sir, who's head of the department, going on organizing for as such as international conferences. And wish you all the best. And as part and parcel of the department, I'm very proud to be one among you all. And thank you. And thank all the participants for being with us on this first day of conference. Thank you. Thank you, sir.
Thank you very much. Now we are ending the meeting. Regarding the feedback, don't worry. The feedback, if you have already responded, it is their record. So we are counting your attendance based on the WhatsApp, YouTube live streaming, as well as on the Zoom also. So this Zoom recording is with there. We are very much with the Zoom recording. We know who have attended really the conference on both the Zoom as well as the YouTube live streaming. So thank you. We'll end the meeting over here, and tomorrow again we will see. Until then, have a nice time.